or should we give people? Yeah. Okay. Thank you everyone for coming to the last day of the conference and the last session. Mainly today we'll be focusing on translation and commercialization of Euglenas. But before progressing, I would like to thank the participants, basically who have attended one, but more importantly, other members of the organizing committee and the organizing partners who have actually made this a reality. So basically, I would like to also thank Scott Farrow. Noblegen, just wave your hand. I'm not sure. Normally, we'll have to wave hand and so people know who you are. And then I have Michael Ginger. Is Michael on the chat yet? Michael? So Michael Ginger, yeah. So University of Huddersfield. Vladimir Hampo. Now Michael is the chair for basic biology, and we had 1 or 2 meetings towards this. Vladimir Hampo, is Vladimir here? Just wave your hand, or yeah, so that people know you. Then Roslo at the Elam Institute, he has also been helping us to do this. So, and we have Alexandra Muro-Clark at the University of Manchester. Alexandra, are you here? So she is with Rob Field. And Christina Pasutova. Is Christina here? Christina. Kevin Tyler, Ecology and Environment. Kevin is not here. University of East Anglia. And then Ben Wagstaff, University of Manchester. And also we also have the supporting partners, organizing partners, Noblegen, represented by Scott, Elam Institute, Neil Hall, and University of Manchester, Rockfield, and most importantly, Emily. Just wave your hand. So Emily has been helping us she actually helped us to put all of these things together in terms of the Zoom and ensuring that we all, are having a good experience online. So someone did ask me yesterday that he's surprised there isn't a subscription for the conference. I said, we have very good partners who have supported this and lots of volunteering time to go into this. So hopefully I don't know if we're having the meeting next year in person or hybrid or online, then we'll decide how that will go. So I just wanted to thank every one of you on this day of the last conference of the last day of the conference. Thank you so much. Then we'll move to the next item. The first, I'm just gonna go straight down to the program. So I'll call on Wade. Is Wade here?
Yeah.
All right. So Wade, yeah, I don't know. You can just give us a wrap up of the meeting yesterday, or did you manage to make it? Because I know you mentioned you. All right. So maybe just a brief intro, and then you can introduce Scott Farrow, who would chair the session alongside Antonio, who will moderate.
Yeah, thank you for joining us today again. Yesterday we talked about the evolution part of the Euglena, and we have 1 keynote speaker and 1 invited speaker, and we also have another session with 4 different other speakers. Today we are going to focusing on the translation and commercialization of Euglena, and our chair is going to be Scott Farrow, and in the very first section, we will have 2 keynote speakers and 1 invited speakers, 1 oral presenter, and then the policy of EIN to make people fully understand how the Euglena International Network is, and then followed up with the Euglenoid Photo Contest Award by a committee of our 3 members. And then the second session will be mainly the discussion of industrial exhibitions, 2 industrial exhibitions. And thank you for joining us today. Scott?
Thank you very much, Wade, for the introduction. And I just wanted to take a few seconds to just thank the organizers of the conference. In particular, thank God, who has made a monumental effort to get this pulled together, and in the establishment of the Euglena network. For those of you who don't know, he's also helping with the Africa Biogenome Project, So we're really happy to have Thank God with us. He's been spearheading a lot of this behind the scenes. So make sure you give him a thumbs up or a thank you before the conference is over. Okay, so we'll have a bit of time this morning to talk about how we're going to translate the research from Euglenoids into commercial applications, etc. And really the future is quite exciting as we kind of are learning as we move forward with Noblegen. And Geoff will speak to how things are going at Kemin as well, but the future is bright. So the first thing that will be on the agenda today is a keynote session speech by our CEO and founder, Adam Noble. Now, Adam wasn't able to join us in person today because he was attending the COP26, where they made a big announcement. Noblegen was part of the big announcement that you'll hear about in our industrial showcase later. But without further ado, I'll start the keynote, which is pre-recorded, and I'll share my screen so everybody can watch.
We're all familiar with these numbers, and I'm not bringing up anything new here, but it is a major problem. The graph is real, by the way. Agriculture is responsible for 26% of our greenhouse gas emissions globally every single year. Our food supply has been designed so poorly, and to add on to this, we need to feed a planet of 10 billion people by 2050. This starts to look like an existential crisis. The time for change was yesterday. It's not enough to come up with new products using the same old agriculture. We need deeper solutions. We need to change the entire food system. And we need to do it with food and ingredients that are not only efficient and sustainable, but are delicious and nutritious. That's where we come in. I'm Adam Noble, the CEO and founder of Noblegen, and today I'm going to walk you through our solution to the coming food crisis. What I am going to show you is a radical elemental transformation of how global food network will work. Thanks for joining me. Before we get into our solution, I wanted to touch on some of the solutions that are out there today. As you can see here, some of the plant-based brands that many of you are familiar with, they are everywhere. The Beyond Meats, the Impossible Foods, the Just Eggs of the world have blown up the market wide open. Every week there is a new plant-based alternative available to consumers, but plant-based meat really does not make a major change to our agriculture system. If you break the process down to its core, you're really just taking a plant-based protein that already exists, like soy or pea, and adding some extra steps to make it more like meat. You never created any new protein, and you've actually just created more waste and more work. Next, we have cellular cultured meat or lab-grown meat. This solution almost takes domesticated farm animals out of the equation entirely. It's real meat that is grown in labs or bioreactors. The science is very cool and the outcomes are truly amazing, but is it efficient enough to change the food system? Will we be able to produce cell cultured or lab grown meat at a scale large enough to offset how expensive it can be to make? I know we will eventually, but for now, we are not. The process just takes too much energy and money at the moment. And unfortunately, you cannot ferment animal cells in the way that you normally do other organisms. When they produce lactic acid, they can die. They're not meant to be outside the body, and they just do not like to grow at that method, and unfortunately creates a significant inefficiency. I'm not going to tell you that these solutions aren't going to make a difference, because they already are. Being able to offer consumers options today beyond meat is a huge milestone and important for the future of any technology. It's going to take many technologies to feed the world by 2050. And there are many companies out there working towards this goal, and we shouldn't discourage any of them. The more solutions we have, the better. The more collaborations we have, the better. We're all doing the same thing and working towards the same goal, and the future of the planet and humanity is at stake. It sounds dramatic, but that is unfortunately the truth. We need to change the food system, and that's where Noblegen and Unite Foods comes in. We're here today at the first annual International Congress on Euglenoids to celebrate Euglena and all the amazing things that it is capable of doing. At Noblegen, we see Euglena as a complex single-celled organism with millennia of cellular intelligence. Billions of years of cellular knowledge can be found within Euglena, and we're working to unlock all of it. We study the natural genius of Euglena and empower each cell to express its own biology, something that we like to call facilitated expression. Since Euglena contains both animal and plant metabolic abilities, it's both more efficient at producing lipids, carbohydrates, and proteins than any of the current organisms in our agricultural systems. This makes sense because the plants and animals our current agricultural system is built around evolved from the unicellular organisms or microbes like Euglena. So if plants and animals evolved from organisms like Euglena, why couldn't we re-evolve the food system from one of the most efficient, adaptable organisms known to science? We think that we can. And we're confident that we can build a completely vertically integrated solution with Euglena. Imagine complete vegan proteins, nutrient-rich carbs, sustainable fats, functional ingredients, and animal and free meat and dairy and egg alternatives, all made from one source at one facility using one technology. It sounds crazy, but that is what we are doing at Noblegen. And that's what Unite Foods is going to be all about. An entire food system made from Euglena. A food system built using a unique mixture of modern technology and ancient fermentation techniques. A food system that encourages innovation and relies upon science. A food system that sees microbes as the livestock instead of the animals. A food system that will work with farmers, not against them. Our goal isn't to completely replace agriculture as we know it today. Our goal is to work with farmers that have generations worth of knowledge to improve the agricultural industry that we rely upon. And if we can do all of that with Euglena, imagine what we can do in collaboration with other microbes, but we'll get to that later. Unite Foods is Noblegen's food and ingredient subsidiary. It's a modern agricultural food brand that is reimagining how the world eats. Unite's sustainable production, smart and scalable design, and all-in-one microbial livestock creates staple kitchen ingredients and food products using the power of Noblegen's microbial platform and fermentation technology. The real game changer to the system is that we offer the philosophy of additive nutrition. Taking something like surplus starch or even organic waste from other industries and turning it into new protein. Absorbing waste and excess and adding a brand new source of protein and valuable nutrition to the world. Our input flexibility allows us to use readily available feedstocks and to customize our supply chain to wherever we are producing our product to minimize the footprint to as small as possible. Our modern barns are efficient and cruelty-free and have zero waste and minimal natural processing. Our end products have great functionality and can be used as drop-in solutions for the pantry staples that would typically be animal ingredients. We offer superior nutrition, clean ingredient labels, complete protein, and immune-boosting beta-glucan, all for the body and all allergen-free and animal-free. The Unite platform offers protein, beta-glucans, and lipid ingredients with a broad portfolio of applications that allow our customers to satisfy the consumers for nutritious, animal-free, eco-friendly ingredients. We have value-added functional ingredients like our texturized Euglena protein crisps, and ready-to-gel beta-glucan. Our egg, dairy, and meat food systems are in development and will be ready to showcase in the coming years. Using a fraction of the energy inputs and leveraging one of the most robust product pipelines in the industry. Our biggest roadblock has always been the sensory and the taste and smell. But over the last couple of years, we found maskers that help, and our product development team has learned how to formulate with our ingredients to make better performing products. The next steps is going to be teaching our consumers how to formulate them into their products themselves. Taste and texture will always be key to the consumer, and if formulators catch one whiff of marine off notes, we found that they don't even try to formulate with the ingredients. If we want to make a difference, we know we can make, our products have to taste and smell amazing. Animal-free products don't have to be mediocre tasting animal analogs. We've already been able to impress the meat eaters with our products, so we know our products can be enjoyed by any and all dietary choices. Studies show that there are plenty of meat eaters that want to become flexitarians. They haven't yet because the taste and texture of animal analogs that they've tried aren't close enough to the animal products that they know and love. But the Unite food systems are making progress, and we know we will have products that fit the mold. In fact, we already do. Our texturized Euglena protein, or TEP, is biomass mixed with pea protein and then extruded by one of our partners in Saskatchewan. We then use the pea protein for protein fortification, but once the protein concentrate has been fully developed, we will use that as the sole source of protein in our formulation. When the biomass is extruded, it comes back to us in easy to use strips or chunks and flakes that can be used in many different animal analog applications. With the TEP strips, we've been able to make animal-free pulled pork and jerky. The TEP chunks are perfect for animal-free chicken applications. The chunks look just like chicken, and when rehydrated with vegan chicken stock, they taste just like chicken as well. We've used them in stir fries, soups, nuggets, and other non-chicken applications like animal-free pork and fish sticks. The TEP flakes are almost identical to flaky tuna. When we rehydrate them with liquid smoke and seaweed flavor, this has the exact same taste and mouthfeel as flaky tuna. We consider the TEP our first animal-free meat analog. Its versatility has given game-changing potential to the industry. Another amazing thing that we have been studying is how Euglena reacts in a three-dimensional growth environment. Here is a video of how Euglena actually looks in that environment. I was amazed when I first looked under the microscope to see what Euglena was doing. In the video, you can see that Euglena is literally assembling into what looks like a three-dimensional scaffolding of tissue. This discovery has major implications for the future of meat analogs. Because Euglena has the same ability to build tissue that animals do, we think that we can work with Euglena to build whole cuts of meat. This same idea is seen in the cell cultured meat, but animals and animal-based products are used, unfortunately, to make those systems. For us, the scale up should be dramatically easier because it is only an extra step or two onto the existing process we have at scale. We're confident to see that we will be able to work with Euglena and other microbes for many aspects of our process, including optimizing feedstocks using mixed microbes to convert feedstocks that we currently can't use. We will also be using the foundation of that structure in other industries. And if we can work with Euglena to master that scaffolding, we should be able to do a lot more than just make the meat analog. We should be able to replicate a number of the same things that have been accomplished with the mycelium technology currently used today: eco-friendly packaging, animal-based leather, sustainable building materials, more eco-friendly alternative solutions across several industries to help reduce the carbon footprint of some of the most significant contributors to the greenhouse gas challenges we have, all made with collaboration with Euglena. In the early stages of my science career, and my search for a dynamic indicator species, I read my first paper on Euglena. I quickly learned just how capable it was, not only as an indicator species, but as an organism in itself. It's safe to say that I became fascinated with Euglena. It may be even safer to say that I became obsessed with it, and I just wanted to learn everything I could about it. I couldn't understand why science had depicted this beautiful creature before me as an outcast. It was one of the first organisms that we were shown under the microscope lens in high school because of how it embodied everything that a cell could be, plant or animal. Euglena just made sense to me. Seeing it as an all-encompassing solutions provider was inherent to me because of somehow it did things completely backwards, but at the same time, it was still able to go from point A to B faster than any other organism could. It wasn't a plant, it wasn't an animal, and I like to think of Euglena as it sat on its own spectrum of dynamic and adaptable functions. In a sense, I think I saw a lot of myself in Euglena because I too thought about things completely backwards. I'm a lot of things beyond this label, but I do sit on the spectrum of atypical. I am on the autism spectrum, and I won't discuss this in depth because I am far more than a clinical diagnosis. Instead, I have chosen to appreciate that this unusual lens that I see the world through is in fact a gift, not a curse. And I have the opportunity to see things in entirely backwards. And without that gift, I wouldn't be standing here in front of you today. For Noblegen, food is only the starting point for us. Every day we work to unlock new practices in production and processing that bring change and help restore the balance of the planet. Science is in our DNA and the backbone and foundation of everything that we do. We view our relationship with Euglena and truly as a collaboration. Euglena has led us to the food and beverage industry just as it will lead us into other industries. In fact, the success we've had working with Euglena has encouraged us to broaden our approach. Going forward, all of the science and technology and R&D that will happen under the Unite umbrella will continue at Noblegen. When key discoveries are made and solutions are found, we will work to see if there's a route to commercialization. And if there is, we will spin off a new company similar to what we have done with Unite. We are looking at programs to sequester carbon into soil, even revamp the approach to modern agriculture at the farm itself. We are now looking at finding solutions for human health, water, biomaterials, energy, mining, and space industries as well. And once we've established our microbial network, we'll be able to offer our expertise and services to companies outside of Noblegen that are looking for microbial solutions. Even if we are building this vast microbial network, that won't stop us from continuing our work with Euglena. Euglena is still and will always be a major part of who we are and what we will do. It's opened our eyes to an entire world of possibilities. Our work with Euglena will continue in both the food and beverage industry and outside of the industry. Euglena got us here, and Euglena will be a big part of the future going forward. Thank you. I'm Adam Noble, the CEO and founder of Noblegen. I'm so excited for Noblegen to be a part of the Euglena International Network, and I can't wait to see all of the opportunities for collaboration as a result of this conference and the future EIN conferences. Thank you to the EIN for having us, and thank you to all for tuning in to my talk. Take care and have a safe time and enjoy the rest of the conference. Thank you.
Okay. Sorry about the video quality. We're going to be posting the video on YouTube after the conference, so you can feel free to watch it again. Thanks to Adam. And I'd like to introduce the next speaker, Ingmar Riedel-Kruse. Ingmar, when you're ready, go ahead.
Yes, quick check. Can you hear me? Can you see my screen?
Absolutely.
Okay, yes. So thanks for the organizing of this nice conference. Very much enjoying it. So welcome everyone. So I want to talk today about three subject areas relating to Euglena. They're all kind of related. First, give some remarks on the phototaxis behavior of Euglena. And you already saw some talk on that by Alan Zhang a day ago. Then significant work that we did on interactivity of how to play and manipulate Euglena. And then I will give some final notes on synthetic biology approaches and the future of Euglena. And you just heard in this other talk what kind of application there could be in the future. Okay, so as you saw in the earlier talk, Euglena is doing phototaxis behavior. It has this flagellum at its front that it uses both for moving forward as well as to turn sideways. It has this eye spot to sense the light. And so basically in the simplest biophysical model, you can just say it's a rod, always moving forward, rolling around its long axis. You have this eye spot, and whenever this eye spot or some sensor in that direction sees changes in light, then it starts turning in his own body reference frame. And Alan talked yesterday about how this, for example, could explain negative phototaxis. So as you can see here, how the cell is basically moving in this helical fashion away from light. Now, here's another movie of this from the top. So you have light from the left, strong light, and Euglena avoids strong light. And you see how it moves to the right, and it's really very obvious to see that sort of behavior. Now, if you look at positive phototaxis, which actually Euglena also executes, so if you have very, very weak light, you see also the cells moving here, but if you look at it, at this particular movie, you will not sense any sort of directionality that these cells have. But if you wait long enough, you actually start realizing they all start accumulating where the light source is. And so this really poses the question, how does Euglena regulate these two different behaviors, positive and negative phototaxis at these two different extreme light conditions? And how does this even work with the simple sensory apparatus and single flagellum that it has? I mean, it has a secondary flagellum, but maybe that doesn't play a role here. So what we know about the negative phototaxis is that there are these two beating states, as Alan also had put out. So you have this rather symmetric beating state, which really leads to this forward helical motion. But if the cell sees a very strong light pulse, then it switches to this other beating state that leads to turning, right? And kind of in this cartoon above here, you see the Euglena is under medium light conditions suddenly experiences strong light and then changes flagella beat pattern and moves away from light. So how could it now do positive phototaxis? And so we have three hypotheses here that we came up with. So the first one is when the cell comes into weak light, that it again initiates the turning direction, but the flagella beat pattern is such, or something else is such, that it just turns in a different direction, right? Instead of turning away from light, it turns now towards the light. The second hypothesis would be that since the light is much weaker, it takes much longer for the cell to respond, about half a cycle, that half a rolling cycle around its own axis, which means by the time it initiates the turn, it has oriented itself such that the turn actually leads to a move towards the light instead of away from the light, right? And the third one, which sounds a little bit odd at first, is that actually the two beat patterns are completely reversed. So if it's in the dark, it does lots of turning, but when it comes into the light, if it sees the weak light signal, then it actually starts moving straight forward with that particular beat pattern. And so we did a number of experiments, and I'm not going into that, but primarily either observing cells under structured light conditions or even holding individual cells with a pipette and then shining light on it and analyzing lots of data. And what we ultimately came up with is that the third hypothesis is really the correct one. So in the top, right, we have this forward movement under medium light turning under high light. But then for positive phototactors is really under darkness, it's turning around a lot, and under low light, if it then sends a little bit of low light, then it actually moves straight. And this actually then also explains the difference in the moving path, so that on a negative phototaxis, you have this almost straight helical path, versus under positive phototaxis, very low light condition, you have this run and tumble behavior. And if anyone in the conference knows more about the sensory apparatus and these sensory proteins and so forth, I would be very much interested to discuss some more specifics of how actually these different behaviors could really come about at a more molecular or subcellular level. Okay, so let me switch to another part of our work. And this was really the idea of, could we interact with cells? We tried with many different cells, but then Euglena really proved to be the top choice because it's rather big. It has a very strong phototactic or photophobic response. And it's also beautiful to look at and easy to culture. And this is work by Sung Lee, who is now a professor at Yonsei University in South Korea. And what you see here is a museum exhibit where a user can draw something on a touch screen, which is projected as light into a microfluidic chip where Euglena cells are, and you can see how these cells, whenever they encounter this light, when they start turning around, right? And this really gives a first-hand experience to visitors to play with microscopic organisms, not just looking through the microscope and seeing them, but really being able to interact with them. And the way this works, kind of illustrated here, the machinery, but in short is that you have a regular microscope, but at the bottom, rather than a homogeneous light source, you have a projector, and this projector is coupled to a tablet PC, and then of course you have a camera, and you really establish a closed loop between all of that, right? And the other nice thing is it's also functional as a regular microscope, so if you look through the eyepiece, you can actually see the cells moving, but you can also see the light pattern that is generated. And that was actually important when we did studies with museum visitors, that people realize this is real and not just a simulation. So here's an example of what this would look like in a museum setting where we did user studies where people could basically play around and then also look through the microscope. And we had prolonged user engagement, so kind of the typical 3 minutes that you want to have in a museum where people discuss and explore. We also use some other interaction techniques, for example, using a Kinect, where we take the whole body of a human and then project that into the microscope. And so people could then interact on a large screen and saw how their own body kind of interacted with these cells. And it's also philosophically interesting because you kind of project yourself into the microscopic world. If you look into the microscope, you actually see a little person dancing in there as light, but then you also bring the Euglena up to human scale. And so this is really interesting of giving a first-hand full body experience with this microscopic life. Other ways of really disseminating that sort of technology is online. So we built cloud labs where you could go online and you had a user interface, as you see here. Most of the screen is basically the view into the microscope. And then we had a joystick here, virtual joystick, which you could pull around. And depending on which direction you would pull, there would be one of 4 LEDs turned on. And so since these cells have this strong negative phototaxis that I talked about before, right, you could really drive these cells into different directions. And so students could explore that, play around. And we did a number of user studies of how students, mostly in middle school, high school, and then also with some universities, how students could learn about biology, but also do scientific inquiry in general, right? Like collect their own data, make models, and learn. And here's a little bit about the system on the back end. So you see many of these microscopes here. So we really had this multiplex so that many students at once could work on these systems. They're all fully automated. Each is driven by a Raspberry Pi and controlled autonomously. At the same time, they are rather low end, low cost. And so you can actually look, if you look through the number and have individual students do an experiment for 1 minute, you can really see how this could scale for millions of students at very low costs, like just a few cents per experiment. And so if this were ever commercialized, this could really be something to make a big difference in education, right? Some other projects we did, for example, using a cell phone. So this is a self-builder microscope, but again, interactive. So you have a little joystick where you can control these 4 LEDs. You put this cell phone on it, and then we programmed some simple games. For example, this Pac-Man here is controlled by 1 Euglena, right? And so you can, with your joystick, drive this 1 Euglena around and try to collect these dots. And as you know, I mean, these cells are partially deterministic in what they do, but they also have their own life. And so that kind of makes for an interesting experience to balance between biological variability and what you as a user trying to do, which kind of gives you a different experience if you play compared to playing a regular video game. And the other nice thing is at the end of the game, you even get some data, some tracing data that you could then analyze in an educational setting, for example. Another interesting effect of Euglena and also actually many other microorganisms is bioconvection. And we also made this in the activity. What you have here is essentially a little flow cell. It's about like 3-4 centimeters wide and a few hundred micrometers thick, a few millimeters thick. And there's about 1 million Euglena in there. And if you project light from below, so you kind of mix the whole thing up and then project an image from below, which you see here, this kind of interesting shape, then what happens over time is that these Euglena avoid the strong white light, move to the black light, and so you can generate these nice images. And then there are other effects in there which are called bioconvection or photobioconvection. Since these cells also swim to the top, the whole when they go away from the light, the whole top gets basically heavy, the liquid gets denser, and then the liquid comes down, and then actually all the cells in there and the fluid on a more macroscopic level starts swirling around and mixing. And so this leads to some other interesting patterning effects. And I come back later. I mean, these sorts of macroscopic controls with light of Euglena might be useful if you think of how to culture Euglena at larger scales or try to sort them. So those sorts of effects might be very useful on that end. But from an interactive experience compared to the one we had before, so this is something you can really see macroscopically. You don't need to have a microscope. And it's also a little bit more robust if individual cells, for example, are not behaving as expected. So this kind of summarizes the different interactive biotechnologies that we've developed in museums, cloud labs, and then also more engineering spaces. Also in classes that I taught, we use this. And you saw also the nice talk from Peter a few days ago, so check out his website. So he really has a makerspace where he brings these sorts of activities to the lay public. So let me give a few final notes on the third topic regarding commercial applications. So we saw in the previous talk that a number of companies are now around these sorts of products to make products out of Euglena. In a general sense, it's something called the 5F. from the approach how to move forward with these technologies. You basically start out with food products, which are which you basically can sell for a much higher price per volume than, for example, fuel. And then the ideas with food, over time you make fiber, feed, fertilizer, and eventually fuel, while at the same time you can kind of scale up the capabilities of culturing Euglena and other algae here, for example, and getting better at the technologies. Right? And if you look at synthetic biology and genetically modification for Euglena, a few things to note is the basic genetic manipulation techniques for Euglena are there, like RNA interference, even all the way down to CRISPR. Of course, we can also do classical breeding, but it's hard, right? It's not as convenient yet and not as established as for some of the model organism. And then also when you grow Euglena, I mean, there's certain things you need to be aware of, that, for example, orthotrophy, that certain vitamins are missing and so forth. And so the question here is whether you co-culture this with other bacteria or further genome engineering needs to be done. Another important, it's also important to say, compared to other algae, it has no cell wall, which makes it easier, certain manipulations and also extracting chemicals. And finally, it's important to note that GMO regulations might apply in certain countries, right? And public perceptions of whether these organisms are genetically modified or not. So that's something important to be taken care of. And so second to last slide, just want to say with our interactive technologies, what we also can do is basically now use light stimuli in various ways to move cells around and then also sort them or concentrate them, which is kind of shown here, for example, with a circle that kind of collapses and thereby concentrates these cells. And so these sorts of technologies may then also be helpful, either on a microscopic scale to identify certain mutants or isolate them, but also macroscopic processing, maybe concentrating Euglena down. So this concludes my talk. So again, I talked about these three different areas: the phototaxis, interactivity, and the engineering. They're all kind of related. I just want to emphasize that Euglena is, as we all know, is really large, colorful, has a strong photo response, and easy to culture, which makes it really very handy for any sort of educational outreach purposes. Also has very interesting biochemistry, as we all know. Genetic manipulations are coming, and so this is something needs to be worked on more. There are huge potential for applications, and the bioeconomy versus the education, ultimately they really need to go hand in hand. Okay, so thank you very much for your attention, and I will answer questions later, I think, in the break. Thank you.
Wonderful. That was a very fascinating talk. Thank you very much. Next up on the agenda is Krishna. Krishna, share your screen when you're ready, and start your presentation. Thanks.
I think Professor Ingmar has to stop.
Exactly.
Yeah.
So
that's all right, no problem.
Yes.
Can you see the screen and hear me well?
Yes.
Okay. Hi, my name is Krishna. I work at the Soft Transducers Lab at EPFL in Switzerland, and I'll be talking to you about some robots I've made inspired by Euglenoids. And this was work that I did when I was at Bristol previously. So as you have guessed from the title, I'm not a biologist. I build robots, and particularly I build robots with soft materials. So things like rubbers, elastomers, plastics, that kind of thing. Here are a few pictures of different kinds of soft robots used in different applications. And you may be familiar with the one on the right, which was in a Disney movie. My particular interest is in locomotion. And this is why I started looking at Euglenoids. And Euglenoids are good at moving in confined spaces. So we all know Euglenoids have one or two flagellum, and then they can move around by beating the flagellum. But when they are forced into a confined space, they exhibit this other type of locomotion called Euglenoid movement or metaboly, where there is a giant change in the shape of the cell, and that is what is driving the locomotion. The picture on the left here is where researchers made a microscopic Pac-Man-like maze, and then they put euglenoids in the maze and put some food particles and predators, just like in the Pac-Man game. And they saw euglenoids switching to this shape change based locomotion strategy. And this was the second picture here you see is work from Professor Antonio and his colleagues, who conclusively proved that this is in fact what happens when the euglenoid is confined. It switches to this other interesting type of locomotion where there is a change in the shape of the cell. Now, I'm interested in this kind of locomotion because it's a good match for the goals that we are solving in robotics, where we want to build robots that can move in confined spaces. For example, within the body, or something moving in a pipeline, or even exotic places like caves on Mars. And so how do we build something like that? Now, one thing you should note is that these organisms, they live in a low Reynolds number regime. So in this regime, the sequence of shapes is important for locomotion, because it's a drag-based locomotion strategy. The sequence is important, not necessarily the speed. It doesn't matter how fast the shapes are changing, but what matters is what the shapes actually are. And if you look at the euglenoid, there's a large change in shape. So we need to be able to capture that kind of large changes in length and diameter. So what we have done is built this pleated balloon-like actuators, which when inflated have a very large change in length and diameter. And what we can do then is combine several of these chambers together to build a robot which can exhibit these changes in shapes. So on the right, you see some of the robots that I've made, which have different scales. And by moving fluid between these chambers, I can get it to change shape like the euglenoid and hopefully achieve locomotion. So that is what I've done. On the left here, you see a video of one of the robots, and it is moving in a viscous liquid to simulate the how to replicate the low Reynolds number motion. And it achieves locomotion like the euglenoid by changing the shape. And because you have a robot, you can do other things which the animal cannot do or the organism cannot do. And you can move in pipes using other kinds of locomotion. Now, the question you might ask is it actually moving like the euglenoid? How do we know it is moving like a euglenoid? So we can use the beauty of maths to come up with a number which says whether the organism is or the robot is moving like the organism or not. So what I have done here is looked at the shape of the euglenoid, or the outline of the shape, and then express that as a set of numbers, in this case, a Fourier series. And what you can then do is you can project this into a new mathematical space where the shape is a point in this space, but this space, instead of being a regular XYZ coordinate space, now has axes which indicate something specific. So the axes are indicating key features that are changing during the euglenoid movement. So as the organism is changing shape from one to the other, what are the key features that are changing? And these are now indicated as my axis. For example, let's say the organism is bulged up on one end. Where is the bulge located on the body, or what is the size of the bulge? And you can do this for the whole sequence of shapes that the organism goes through during the cycle of motion. And what you will notice is that it forms a kind of like a loop or a circle, let's say a loop in this new mathematical space. And you can do the same thing with the robot. And now you have two shapes in this new space, and you can compare how close these two are to get a number which says, oh, my robot is, for example, 85% similar to the orgasm. So this is a technique to compare dynamic shapes, and this is how you can conclusively say that, oh yes, my robot is actually moving like the Euglenoid, and it's able to capture, say, 85% of the shapes. Now, as a biologist, you might look at this and say, oh, hold on a minute, Euglenoids don't move like balloons. They move by deforming the pellicle. So what the Euglenoid actually does while changing shape is something like this. On the left here in the picture, you see these lines. So these are these indicate strips on the surface of the Euglenoid, which consist of these S-shaped cross-sectional strips, which are interlocking with each other. And there are microtubules between the strips, and the Euglenoid changes shape by sliding these strips. So the right here is a little crafty illustration of what that looks like. So if you have a cylinder with lots of stripes, and then you open it up and flatten it out. Now, if you take each strip and move it a little bit further with respect to the previous one, you end up with this thing in picture C. And if you roll this up, what you end up with is a shorter but a wider cylinder. Now, the Euglenoid is capable of doing this locally in any direction it wants and to any extent it wants. So that is how it is getting all these various shapes you see here. And what these pictures here show is how much the relative sliding is between two neighboring strips at each location along the strip. So we can use this information and fabricate similar strips. So on the left here, on the top picture, you see a microscopic cross-section of the pellicle in the Euglenoid. And on the bottom is strips that I have made with polyurethane, which mimic this interlocking S-shaped design. And you can create a surface of these strips, and they slide against each other. And in this slide, what I'm showing you is you can change shape by deforming these strips. So you can slide them against each other just like the Euglenoid is doing, and then you can get a new shape. So you can both have it happen passively. So you apply an external load and the shape is, the strips conform to the load. Or you can also actuate the strips to move. So in this case, what I have done is I have the spring-like structures you see here are coiled polymers, which when heated contract. So that is how they move strips against each other. So you can realize shape change just like the Euglenoid is able to realize, but in an artificial system here. There are a few challenges to realizing the whole potential for changing shape drastically like the Euglenoid using these artificial pellicles. And one of the main challenges is that we don't yet have an artificial system which works like a biological muscle at the scales that we want, for example, at the micrometer or the millimeter scale. And it's very hard to implement localized sliding and to supply power to these localized points. So I would like to conclude my talk saying that I've taken inspiration from Euglenoids to design a few different kinds of new locomotion strategies and shown that it can work in soft robots for locomotion, and it can work at a few different scales. And we also developed this artificial pellicle-like surface, which can lead to a shape changing or a shape morphing design if you want to use it in such a way. And if you have any questions, you can contact me or ask me later, or you can also contact my advisors at Bristol who I've worked with.
Thank you very much.
Thank you.
that was a wonderful presentation. Very interesting. That you replicated the helical. Okay, so the last speaker on the panel session today is going to be Geoff Horst from Kemin. So Geoff, when you're ready, take over.
Thanks, Scott. Can everyone see my screen right now?
Yes.
Okay. Well, thank you very much to the conference organizers. It's been highly educational, and in fact, I've got some notes here from today's talks to try to get my kids' science teachers plugged into these Pac-Man games. It sounds like a really cool opportunity to really bring some tech to the biology and vice versa. So today, what I'll be speaking about is how beta-1,3-glucan works in the immune system. And so I think if anything, over the past 2 years with the pandemic, we've all learned more about immunity than we ever probably expected or necessarily wanted to. And I think overall it is good for us to become more educated as just humans on how the immune system works and how it can help protect ourselves from both COVID and going forward. So, so just a quick background on the immune system. So we actually have two parts of immunity. We have the acquired immunity, and this is what takes advantage of those antibodies. And so you can develop these antibodies either by being exposed to a pathogen. So if you were sick from some disease years ago, your body still retains some of those antibodies. So then if you see it again, your body can respond very quickly to that same specific pathogen. Or we can actually get antibodies passed from our mothers. And then, of course, there's the vaccine approach. And so we can actually give specific antibodies to both animals and humans. And that helps to trigger this specific type of immunity, which is very efficient and it's very fast. So if you have these antibodies, the immune system can respond really quickly and targeted. The other part of the immune system is the innate immunity or non-specific immunity. And this is more of a generalized approach. And it's specifically, it's intended to be non-specific. So this is what happens every single day as we encounter all sorts of potential nasty pathogens that are in our environment. So whether it's viruses, bacteria, parasites, whatever, this is kind of the first line of defense, and it takes advantage of all sorts of different parts of the immune cells. So this is macrophage cells, natural killer cells, and a lot of our innate immune system is in the gut. That's kind of the front line of how our bodies interact with the outer world. And so we eat tons of bad stuff every single day that's on our foods, but we don't get sick because the innate immune system works usually quite well. And so beta-glucan, which I'll show, is actually it's an immune modulator, and it can actually help in both forms of this immunity. So what is beta-glucan? It's simply a polymer of glucose molecules that are arranged in a very specific pattern. And so beta-1,3-glucan just simply means that the glucose is attached at the 1 and 3 carbon, but there's other types of glucans. So cellulose, the most common polymer in the world, is beta-1,4-glucan. Starch is an alpha-glucan. So similar structures, but they're actually arranged in a different way. Now, the fact that beta-1,3-glucan is found in the cell walls of potentially pathogenic organisms like yeast and bacteria, what's happened is that over time, animals, including humans, have sensed this specific molecule and this pattern with being sick. And so now our immune systems recognize that when we see beta-1,3-glucan, it thinks, hey, something might be up. There might be a potential pathogen in my body, and I need to do something about that. And so this particular molecular pattern, the 1,3, is pretty important because our immune cells have these receptors, and you've probably heard of the key and lock approach. So there's these immune receptors on the immune cells that all they do is recognize that 1,3 pattern. Now, if they see 1,4-glucan, which would be cellulose, they think, oh, no big deal. That's just something that's food or it's inert. Or if it sees starch, which is alpha-1,3-glucan, it's thinking, okay, I know that that's a food, so it doesn't respond negatively to it. So it really is the specific 1,3-beta-glucan that our immune system is responding to, and those of other animals. And so researchers decades ago actually recognized the potential to use beta-glucan or beta-1,3-glucan as a tool in how we fight diseases. And so there's been dozens of different yeast-based beta-glucan products that have been brought onto the market, both for animal health as well as for human health. But the challenge is that extracting it from yeast cell walls is relatively difficult, that it's expensive. And so what we decided well over 10 years ago is maybe we can use Euglena, which produces beta-1,3-glucan beautifully as these pure granules, paramyelin granules, which are 100% pure beta-glucan. Maybe we could actually produce that target molecule in a much more efficient way that will allow us to actually get it into the food chain, especially for animals, at a much lower cost, so that its adoption could be more widespread. So that's what the original company, Algo Scientific School, was, and we were later acquired by Kemin here. And so here's just a little bit more detail on those very important paramyelin granules. During our research, we figured out ways to fluorescently label them, which makes it cool for doing some microscopy work. On the right-hand side is the scanning electron micrograph of a paramyelin granule. And you can see these little strips, or these kind of ribbons. Those are actually the triple helix of the beta-glucan. So the beta-glucan is hundreds and thousands of glucose units long, these long strings that's arranged in this beautiful triple helix pattern. And you can imagine these immune cells, they're interacting with the outside of this fuzzy particle, and so that's where they're detecting that 1,3 beta-glucan pattern. And so when you're trying to use this molecule to the advantage of an animal, the goal is really just to prime the immune system. We don't want to hyperactivate the immune system, because that's just going to cause inflammation in the animal. And if you have an inflamed gut, that's not going to help you digest your food very well. And as we know, inflammation overall is not healthy. So what we want to have is a balanced immune response, and that's what we would call priming the immune system. And I'll explain a little bit more about that. So the mode of action of how we use a product that's including Euglena beta-glucan is that we feed it to an animal, or you can take it as a human as a supplement. The beta-glucan granules are going to go down into your small intestines, and that's where it's going to be actively captured from the intestinal tract by some of the immune cells that are actually found in the small intestine. So they're going to bring those beta-glucan granules into the body, and then other immune cells also start to consume and phagocytose those particles. And then the last step is really the priming signal. So this is where the immune cells are going to send out chemical signals to the rest of the body saying, hey, we think we found something, we need to pay attention here. And so I'll illustrate a couple of these with some pretty pictures. And so here's the first one. If we feed mice and other animals these fluorescently labeled beta-glucan granules, we can see that if we sacrifice those animals and look in the intestines, you see here some villi. So the intestines are covered in these villi, high surface area bodies that are covered in immune cells. And we see that they're capturing these fluorescent beta-glucan granules from the lumen, or basically the intestinal tract, and pulling it into the inside of the body. So that's the first step. Are they actually being actively captured? And in fact, they are. And then the next step is to show, okay, these other immune cells like macrophage cells that do a lot of the heavy lifting for the innate immune system, they can recognize these beta-glucan granules very quickly. So on the left-hand side, we've incubated these macrophage cells with the fluorescent beta-glucan granules. And within less than 2 hours, you can see here that they took in almost like 20 pieces of those paramyelin granules very, very rapidly. And the right-hand side shows a macrophage cell, which is that giant blob in the middle with the blue arrow. It's starting to encounter one of these paramyelin granules. And then a little while later, you can see that it's starting to phagocytose that particle. And so they recognize that pattern. They say, this doesn't look right. I'm going to phagocytose it. And then they've already taken in a few other beta-glucan granules at the same time. So there's like 3 or 4 other ones in there. So that's that recognition step. And then we actually start the real fun, which is, okay, how do we alert the rest of the immune system? So at different dose levels, we elicit stronger and stronger responses of these chemical signals. So we have the pro-inflammatory cytokines. These are the ones that say, hey, I found something. We need to bring a lot of attention to it. But it needs to be balanced with the anti-inflammatory signals that says, yes, we hear you, but let's not go crazy here. We don't want to cause extra inflammation. So if you have both of those going up at the same time, it still has a balanced response, but we're priming the immune system because it knows something is out there that we don't know, and we need to recognize what it is. And so the last chemical signal is these chemokines, which is kind of the call to action saying, hey, let's bring more of the troops, especially to the gut, because we want to be ready so that if there's a lot of these pathogens coming, we want the army sitting right there and ready to go in case there's more of them than us. And so that last component, which is recruiting more of the immune cells to the gut, is kind of illustrated here. Again, this is with mice that we've been feeding beta-glucan. And here's the villi again. And this is where we, the higher fluorescence segments on the bottom panels show that we've recruited more of the immune cells to those villi. So now we've got the army sitting there right at the enemy line, and it's not causing inflammation. So it's not causing stress to the animal yet, but it's there. So then if, let's say, a salmonella pathogen makes it through, or some virus pathogens get through, the army is there and ready to fight. And so that's what's basically happening when we feed beta-glucan to animals. And so during the expo session in a little while here, some of my colleagues from Kemin are going to describe the various products that we have already on the market out there. Alita is the product that we developed over 10 years ago for animal health. And then Beta Via is the supplement that's used in the animal or in the human health market. And so the power of Euglena used to our benefit. And so I'm happy to take some questions when we have the panel discussion here. And also my colleagues will be happy to take some more questions later. So thank you.
Thank you very much, Geoff. That was a great presentation. So before we move on to the panel discussion, it's customary here in Canada at the 11 o'clock hour to have a moment of reflection for Remembrance Day, to recognize, or Armistice Day, to recognize the end of hostile World War I and World War II. So if we could take maybe 30 seconds to do that, that would be very much appreciated. Okay, thank you very much for accommodating that. Okay, so for the panel discussion today, feel free to drop your questions into the chat box here. I will field them as I see them, as the order in which they have appeared. So why don't we start with get a little more clarity on Thank God's question, which is to Mark Field and Michael. You have some of this work on the sensory apparatus or iSpot. Maybe you can comment and elaborate a little bit more, Michael Hammond or Mark Field.
So I think this would be Michael Hammond. Michael Hammond is here today.
Yes, I'm here. Can you hear me fine?
Yep.
Yeah. So there have been a series of studies over the past 20 years looking at light-sensitive proteins, which we localized to the flagella in our flagella-related proteome study. And these are proteins that coordinate phototaxis, which is this photoactivated adenylate cyclase complex, as well as other PAC-like proteins, which also mediate the step up and the step down response, which is an increase or decrease in swimming speed depending on light intensity.
Okay, wonderful. This
You
question.
Do you know anything of whether the positive and negative phototaxis is most likely done by the same sensory apparatus, or whether there are two completely different sensory proteins might be there to mediate?
The research that I've done suggests that it is coordinated by the same protein complex. So you have, PAC alpha, PAC beta, and they're arranged in this complex on the paraflagella body. And they found that mutants that don't have this paraflagella body are incapable of performing either positive or negative phototaxis.
Okay. I think it would be very interesting to have, I don't know, 10-15 minute chat together. I also want to say like the step up and step down response, which are all over the literature, which is a more macroscopic look at, where you also set like different velocities. it's not clear whether this is really true on a microscopic level, because you can have a lower velocity if you swim like this rather than that, right? So there's certain questions about measurement and definitions that we try to piece out. So thank you.
Okay, next question goes to Ingmar. Thank you for your impressive talk. One question: do you have any concrete evidence for which one of three mechanisms is correct for positive phototaxis?
Yeah, so I mean, the first two with the delay and switching in a different direction are definitely not correct. So we have evidence that that can't be true. All evidence that we have clearly shows that the last one is true. But as you know in science, right, like you can test so many things, but maybe someone else finds something else. And we couldn't think of a fourth hypothesis. So it really seems to be that it does at very low light, is doing this run and tumble. And really this also relates a little bit to the what does step up versus step down mean, because it's also kind of an inversion of if the light intensity increases, from which state to which beating state do you switch? So, but yeah, we think it's the third one.
I guess related to that, has the phototaxis action spectra been determined? Assuming that means the wavelength, specific wavelength, or?
So I know there are a number of papers out there that really measured 4 different wavelengths that the cells respond. For example, to red light, they don't really respond much. But I mean, it's broad. And I also know that I think there are 2 different peaks. I don't have the literature in my head right now, but there's a number of literature out there kind of looking at the response for different wavelengths of light.
Great. For Krishna, at the million micrometer scale, could you start with existing molecular machinery and work backwards rather than trying to create them from scratch?
I would say it may be possible, but I would say it is challenging. The reasons I think are as you scale up, you would have to see how the force scales with size and whether or not that is going to give you any meaningful actuation or not. The other thing is if you want to say use something from a lower scale and you want to assemble it into a structure on a higher scale, how you assemble these motors, for example, matters, and it's not easy to for example, take a molecular motor, put lots of these and then get an actuator at a millimeter scale. The other thing is that some of these motors, they work in specific media, and it may happen that the robot you design only works in that specific media if that is the only media in which your motor is going to work. But if you want to use it in an application where you cannot have the energy source from the media, then I think it may be a challenge.
Wonderful. And here's kind of a cool question. So crazy idea. This is from Emma. The knowledge gained from your robots based on this Euglenoid movement, do you think it has any applications in the design of less rigid stents to correct vascular obstruction?
Mm-hmm. Yeah, I wouldn't say it's a crazy idea at all. It's totally possible. Yeah, one I can imagine something maybe moving up an artery and then deploying somewhere to ease the obstruction. So I think it is totally possible. I would comment though that I think the issue was with rigidity. So you can so the softness is a very vague term, I would say. So you don't necessarily have to use soft materials to have a soft stent. You can use a rigid material like a metal or any other rigid material, but then arrange it in an interesting design to make it behave soft. So I think there's interesting design both in terms of the structure and the material and also the actuation.
Yeah, because I'm Emma Saavedra from Mexico. Because a problem with the stents is that the rigidity causes inflammation. and it causes a rejection of the stent. So it is very frequent. So in a more soft and soft material or soft adaptation to the vascular lumen, it could improve the function of the stent. Thank you very much.
No, thanks for the suggestion. It's something good to think about.
I have another question. This one's for Geoff. Could you expand or just go back to the comparison between yeast and Euglena? I think maybe, thank God, we're talking about paramylon or beta-glucan, the differences. Is that correct?
Yeah, and so back before Euglena could be produced commercially and cost effectively, the only good source of beta-glucan was from the yeast cell walls. And so a yeast cell wall, it's very rigid, obviously much different than Euglena. And the beta-glucan is actually like the rebar that kind of holds the cell wall together. So it's a pretty stiff and tough structure, that yeast cell wall. And so, in order to get the beta-glucan out of that cell wall and purify it so that you can have that very important exposure of the 1,3 beta-glucan pattern so that the immune cell can see it, you have to purify it enough that the immune cell is going to see a clean pattern. There's also a slight issue that the beta-glucan that's in the yeast cell wall also has these side branches. So it'll look more like a tree where the actual sticks are the 1,3 beta-glucan stick, but you have these side branches that come off. And so then you can imagine that the immune cell receptor, it might not get a clean look at the piece that's actually the beta-1,3 glucan if it's, clamping in on, let's say, one of the branch points, because then you have 1,6 and 1,3. And so the Euglena has the advantage that over 90% of the beta-glucan in there is just this very nice linear beta-1,3. So there's, huge amounts of linear space for the immune cell to key onto it. And when I mentioned that these, strands are hundreds of glucose units long, that makes it an ideal format for those immune cells to see it. And in fact, getting a little bit more complicated, the immune cells actually have multiple receptors, of course. And so we think that it actually requires you to bridge multiple receptors at the same time to actually turn the immune signaling on. And so having that nice clean structure, like a long straight stick as opposed to a branchy bush, makes it a little bit more of a cleaner molecule and format for the immune cells to respond to. And at the end of the day, it all comes down to the cost. We need to make it cost effective to use in chickens and pigs and fish, and it just wasn't possible with the yeast platform. So thankfully, Euglena is the world's best organism that we know of to make beta-glucan right now.
Thank you. That's great clarification, Geoff. Not all beta-glucan is the same. Yeah. Okay, wonderful. Well, if there's no other questions, I think we should move on to the next part of the session, which is going to be a presentation by John Knight, our EIN Euglena Network Coordinator. So I'll hand the floor to John, and John, when you're ready to share your screen, please do so.
Yeah, so let me just put a context to John's presentation, if that's okay, Scott. To some of us who may not know or who have been following the development of the Euglena International Network, we actively started to work on this network way back in February, basically. That was when the active development started, but it's something I've talked about for a while. Then it has been mainly focused on volunteerism from members of the executive committee, steering committee, and advisory board. So far, what has happened has focused more on awareness creation, organizing conferences, formulating policies, and trying to understand what to do. But as the network picks up and gains momentum, people come on board, and things are beginning to be clear. It now requires more active engagement by dedicated personnel and potentially institutionalizing the Euglena International Network, mainly to ensure sustainability, both in the short term, medium term, and long term. So one of the things we try to do is to have John Knight, basically, and that was mainly due to its support from Noblegen as well. Basically, so the idea is to for us to get a document, you know, and also understand what framework we need to work on, and who would be the partners, who, you know, how can all of this happen. So I'm sure John will talk more about this. So, but that's just I just wanted to give a background, a context to this, particularly because there is now need for us to institutionalize in, and it's not just driven by volunteerism. Of course, there will be volunteerism from the science part, and, you know, we'll still have a science committee, but we now need to start thinking about that, and that's what John will talk about today.
Perfect. Thank you. John, whenever you're ready to share, go ahead.
Is that coming up yet?
Yep, there it is.
All right. I don't have it on my my
There
screen.
it is there it is at the top, John. Just click on the tab on the top of the Chrome browser to your left.
Top left.
The tab, looks like the tab is at the top there on your Chrome, sorry, up. Just at the top of your screen, I think I saw the tab on your Chrome browser.
I just can't seem to get it up. That's not the right thing to say either. Do you see anything at all?
Yes, just click it. Click that one. Click the window. Yes. Then click share.
So I've got a brand new Chromebook here, so I'm just trying to. Sorry for my ignorance here, and then hit share.
Yeah, click hit share. Yes. Thank you.
And then it looks like the tab on the top. Yeah, there you go. Perfect.
There we go. Ah,
Thanks.
Thanks very much. Thank God you just pretty much gave my presentation away, but I appreciate the introduction. I'm looking forward to this opportunity, and I just want to share some thoughts on the steps that we're going to take in the organizational evolution of the Euglena International Network. I really enjoyed yesterday's presentation on the evolution of euglenoids, particularly the comments or the debate about whether parasites had a role. I have to say that I don't think parasites will be involved in the organizational evolution of the Euglena International Network. At least I hope they won't be. But anyway, so what I'm going to talk, we have right now a very informal but organized scientific and industry activities that are taking place. I mean, there's committees that are set up, there's a website, there's member recruitment, there's this conference is an example of activities that are taking place already. And I want to move into what formalization means. It means just taking this group of volunteers around a common interest or cause and create an official organization, call it an association. In a way, it resembles a club. And eventually it could lead into more formalized structures such as a nonprofit organization. Some of the benefits, well, and a formal organization can make official agreements. That's the major benefit. And I think that we're going to need to have agreements in the future with partners or strategic alliances, with members, and also having a formal policy and procedure document will help with letting people know what the EIN is about and what it's doing. What needs to be done is basically creation of that document, creation of some of the fundamental agreements that we're going to need with partners, and discussion with the members as to what the organization is moving to. Timing will be the next 11 months. One question left out was why, and out of the benefits section. And the basic reason why that we need to have a look at formalization of the EIN is the change in people over time. And that's fairly common with any organization where people change over time. And the institutional memory changes with the people. So you can imagine what happens when this guy leaves to the organization. Thank God has done a tremendous job. His energy and dedication has been tremendous in what he's done and bringing this organization along. But what happens if he leaves? And he doesn't intend to leave. In fact, there'll probably be a presentation a few decades down the road where this same picture will be put up on the screen beside another picture of the current, thank God, with white or gray hair and beard. So you can imagine that into the future. So right now we have a good established organization through the efforts of volunteers. But what's next is actually writing it down and writing out the structure and writing out all the policies and procedures that the organization is going to follow, which can lead to programs, promotion, and maybe down the road advocacy. Ultimately, one pathway could be that the EIN could be a self-funded nonprofit corporation. Our guiding principles. These are the three, there's many guiding principles, but these are the three primary ones for the formalization process that we're keeping in mind. Want to maintain our core, which is our heart, is our scientific research and our international group of researchers that are making new discoveries about euglenoids and also finding real-world applications of these discoveries. We want to build global awareness about Euglena research. We are a small organization, but we gain strength and value with our expertise around Euglena and our international network, which reminds me of, I came from a university environment with a very small university, Trent University. And at that university, we had tremendous success in Canada in fundraising from government and private partners because we did two things. Small university, we did two things to take on the big guys. One thing that we did was we focused on what we were good at in research and supported our faculty that worked in the environmental areas. And the other thing we did is we brought in and broadened our partnership with government, municipalities, industry partners, foundations, and had a very broad network of applications for our research. And that's gave us extra strength. And I think EIN can do the same thing. This is a little bit scattered, but it's the basic elements of the formalization process. The two yellow that's the very basic written documentation and structure of the organization. We have partners with strategic alliances or with industry partners, and we have members. Are the three areas that the formalization documents will deal with. So lens number one is the starting point. Policy and procedure document, the structure, how our governance will be of the organization will work, how our leadership will be selected and monitored and guided, having a memorandum of understanding with founding member partners who will be hosting initially the EIN, and that's the starting point that we're going to work from as we expand. The second element is actually the types of agreements that a formalized EIN can sign with external parties. And we use the memorandum of understanding again, with our strategic alliance partnerships, but also we can also have agreements with our members, whether it be individual researchers or research institutions and industry partners. And the document that we'll be preparing will have different categories of membership, which is important because the third lens that we're going to look at is how is the organization going to be funded over time. Industry partnerships are going to initially play a critical role, but over time, as the organization grows and resources are required, I think over time there's going to have to be membership charges, and probably with the categories of members that we will establish, will have different rates of membership fees. So this is, I don't have a picture of me licking my finger and setting it up in the holding it up in the air to see which way the wind is blowing with this comment, but I'm sure I'll get some feedback from members as time goes on as we prepare this document. So that's the short presentation, and please feel free to contact me at that email address, or connect or follow me on LinkedIn if that is okay with you. Thanks very much.
Thank you very much, John. Very exciting progress to look forward to. If you could stop sharing your screen, I think we're going to move on to the next part of the session, which is an exciting addition to the program, and that is...
Sorry, Scott. Is there a chance we could just ask verbally if anyone has any comments based on this to John's presentation? It would be nice to maybe spend like 2 minutes trying to gather feedback. Is that okay with you, Scott? And see what people think. Sure. Yeah. So just briefly. So I think I can see Barry Seville.
Yeah, thank God. Thank you. One of the things that I can bring to the table on this is having been the president and being on the executive of other scientific committees that have filed for and received not-for-profit organization status. It's going to be important to come up with a finance stream. And I agree with John that we're probably going to be looking at membership dues at some level. But one of the major things that has driven funds for various societies I've been involved with has been profitable interactions with scientific journals, and having establishing a journal that you could facilitate through the society that we develop. So there are various ways to get money, but that one has been quite beneficial. Also, you can raise money with conferences. And what you would think about spending this money on then is organizing the conference, because quite frankly, if you're going to bring people in person, you have to put up money to get a hotel reserved, and it'll be thousands of dollars, right? And you have to have all of that sort of stuff to reserve the hotel, etc. So that's why you would need the money. The other thing is that you would want to have money, presumably, to encourage students, right? By giving them awards for talks and presentations and stuff. So just some random thoughts, thank God.
Thanks, Barry.
Yeah, thank you, Barry. I think I can see Ingmar was mentioning what would the money be used for. So for instance, like this particular conference, we didn't want to charge any fee because first of all, we want to be able to finalize the hosting location one. I also want to be able to be a formal organization, and most importantly, have a treasurer that will manage all of these accounts. So the idea is to run it without any registration or anything for like 2-3 years until such a time when all of this have been sorted out and we know there is a platform, to, someone that can manage all of this basically. So I think Ingmar, I don't know if that answers your question. There is quite a lot. For instance, the Zoom account, I think with the specials we used yesterday, it was Scott Noblegen. With the Zoom account, it was Airline Institute, and also there are so many other things, including the website. I think with the website, it was me that had to do that because it was just a small. So there are quite a few things here and there. It's not such a huge budget for now, especially with most of the activities happening remotely. But beyond, from 2022, we need to start thinking about how to formalize things and institutionalize things, basically. So Scott, back to you. So if there are any more questions, please feel free to reach out to John or myself, basically. Thank you.
Wonderful. Good discussion. Thanks. Okay, so if we can move on to the next part of the session, that's the Euglenoid photo contest. And I'd like to call the committee members, Mark, Takahiro, and Rosina, to make that presentation if they're able to do that. Is Mark, Takahiro, or Rosina here?
Yeah, so Mark did send apologies that he had another meeting. So I'm not sure if Takahiro and Rosina, are you here?
I see Rosina in the
I
crowd.
am here.
Yes. Okay. Do you want to just comment on the nominations for the photo contest?
Yes, we have a lot of discussions about the participants in the photo contest. So we conclude to nominate in the first one moment, please.
So maybe while you do that, I don't know, Takahiro, do you have any comment? I mean, do we have a winner? How did it work? How many people submitted? And why did you choose the winners? I mean, those points basically. I think Mark was supposed to do this, but I'm not sure if he sent out an email to you guys. But he just sent an email to me this afternoon.
Yeah, we choose 3 winner. I'm sure. And so in my criteria, so I choose a picture based on the very likely Euglena. And unfortunately this time, so there are not so much applicants this time. But so at least we choose the 3 winners. And sorry, I can't find the name.
Yeah. So I'm not sure how I want to do this now, because the idea is to have top 3 and then have the top person. So with Mark not being here, so I don't know, did you guys get the email from Mark? I mean, did he? So maybe we can move forward with this, Scott. Then much later, maybe I can have Mark send an email around on what the nomination is.
Let's postpone it. And the good news is we know there's a winner, and that winner, the suspense will just be held a little bit longer.
Yeah, so I would have Mark send an email to the winner. So I'm not sure how I want to do this now, because I was thinking Mark will be able to announce the winner here. What do you think?
I think if we don't know who the winner is at this time, maybe we can postpone until this afternoon's session, and maybe we can get in contact with
Mark. All right, perfect then. Okay.
Okay, so let's move on. So that concludes this morning's session. We have a little bit of time for a break. It was scheduled to go to 16:35, but we're already approaching that hour. So why don't we, is everybody okay to take 5 minutes maybe? We want to take 10 minutes, 5-10 minute break. Is that okay? Meet back here at, let's say 16:45. Is that okay?
Yeah, that's fine.
Okay. Okay, let's meet back at 16:45, 10 minutes from now. Thanks, everybody.
I found the email from Mark Field. So I'm just going to forward the email to you. Let me see, just 1 minute. I'm going to forward the email to Rosina. So because I think this should ideally come from, they are the members of the committee, so they can check their nominations and then say who the winner is. Is that okay?
You want to do that at the end? And
okay, at the end is fine. So Rosina and Takahiro, are you there? Yeah, so I will forward Mark's email to you. Then you just look at the top 3 and then announce the winner, basically.
Okay.
All right.
Okay, so I'm going to hand over the mic to Antonio, and he's going to lead the second session here.
Very well. Welcome to the final session. And so we have a first presentation by Kemin Industries. And Geoff, can you share the screen and the presentation?
Yes, thank you, Antonio, and thank you, everyone. I'm going to first introduce the other Kemin team members. So we have Brenda, Penny, and Chris. And so they're joining from various locations across the country. Do you guys want to say a quick hello?
Yep, just had to unmute. I also have a coffee cup that says you're on mute. So it's been fun. Really nice to join the conference. Excited to be a part of this and share with you a little bit about our corner of the world with our Euglena.
Hi, and I'm Penny Woods. Chris Sadewasser and myself are located here in the Des Moines office.
Yeah, thanks for having us. Appreciate it.
Okay, so I'm going to start off by sharing a video from my computer. It's about 20 minutes long that has a couple different introductions to Kemin as a company, as well as our Euglena products, and then we'll have plenty of time afterwards for a Q&A session. So I'm going to attempt to start the video. So wave on your cameras if it's working or not working, like thumbs up or thumbs down. So here we go.
We are scientists, inspired by the world around us. We are visionaries who see things that others don't. We are innovators and farmers and researchers. We are moms and dads, the parents of tomorrow.
Every day, Kemin team members are here to add value to the entire value chain. They bring home to our customers the type of nutrition they expect.
We are Kemin, and we are transforming lives. Founded in 1961 by R.W. and Mary Nelson, Kemin Industries began modestly on a family farm in Des Moines, Iowa. Today, however, Kemin has grown into a global ingredients company, improving health, feeding a growing population, and ensuring the safety and well-being of billions of lives. In more than 120 countries around the world, in Europe, the Americas, China, Africa, and Asia, in laboratories, in fields, on farms, and in factories, Kemin Innovation is finding solutions to the world's greatest challenges.
Scientific discovery is based on having and testing hypotheses. If what happens is different from what you had expected, that doesn't mean the experiment was wrong. It means you have a possibility of a discovery.
Built on exacting science and powered by human imagination, Kemin works to sustainably transform the quality of life every day for 80% of the world with its products and services.
The quality and safety of our ingredients is absolutely critical. If just one mistake makes it to market, our customers are at risk. That's why we test, retest, and test again. Our clients trust us.
Kemin works closely with our customers to increase an awareness of how their products interrelate with local environments, food safety, and nutritional demands. In fact, every Kemin facility around the world has a customer laboratory services team dedicated to helping our customers with their unique challenges.
For over half a century, Kemin has applied science to improve the quality of life. We embrace the exploration of new areas, finding unique solutions to changing needs. We truly work with an eye on the future, developing scientific solutions today that will be ready to benefit you tomorrow.
We are relentless in our pursuit of a better world. And because we hold ourselves to a higher standard around the globe, you will find dedicated professionals with a deeper commitment, a passion for what is possible, and a desire to do more for the communities we serve. We are Kemin, and we are sustainably transforming the quality of life.
Hi, my name is Penny Woods, and I'm the Marketing Director for our Kemin Human Nutrition and Health Business Unit, and I'm honored to be here today with two of my colleagues, Brenda Fonseca, who is our Global Technical Service Lead, as well as Chris Sadewasser, who is our Technical Service Manager. I would like to introduce you and give you a little insight into our organization. This is Kemin. At Kemin, our vision statement matters. We believe that by striving and serving our customers, that we can make a positive difference on billions of people's lives around the world. We strive to sustainably transform the quality of life every day for 80% of the world with our products and services. This growth in the face of limited resources will present daunting world challenges for people and animals. We are inspired to discover new molecules and breakthroughs to help solve the challenges and improve the lives of billions. Kemin has more than 15 manufacturing locations worldwide, and we operate in more than 90 countries and have customers in more than 120 countries. Every day, we strive to sustainably transform about 7 billion lives with our products and services. And we do this through exacting science and technology and offering more than 500 specialty ingredients to customers. Kemin was founded in 1961 by R.W. and Mary Nelson. The company remains a family-owned and operated organization with more than 500 patents and applications and totaling more than 900 million in annual revenue. The first plant was located in a wool warehouse. The current Maury Street visitor parking lot is where R.W.'s father's house, where his father housed sheep. R.W. and Mary still work in the office and remotely every day and travel around the world for business. Dr. Chris Nelson is our president and CEO, and Libby Nelson is our general counsel and vice president. The first Kemin facility and corporate headquarters was located in Des Moines, Iowa, where it is still located today. Today, Kemin has grown globally to include manufacturing facilities in more than 15 countries and serves customers in 120. With the workforce of more than 2,800 employees, the manufacturing locations, research facilities, and offices outside the United States, we meet the needs of our customers worldwide. Kemin continues to invest in regional headquarters, laboratories, and manufacturing facilities around the world. The blue dots on this map represent Kemin's major manufacturing facilities. The red dots represent additional sales facilities around the world. The purple dots represent locations where Kemin has facilities to grow and harvest our specialty ingredients. Kemin puts the future in focus. As a responsible family-owned and operated company, Kemin is committed to sustainably improving the quality of life now and for generations to come. This commitment is at the heart of our servant leadership approach, which compels us to focus on the growth and well-being of our employees, our customers, and the billions of people that we serve every day while safeguarding our planet's finite resources. Kemin will magnify this commitment to sustainability using a model focused on people, our planet, and business. At Kemin, we view sustainability through 3 lenses: healthy people, healthy planet, and healthy business. It's where these lenses overlap that we find our actionable pillars. And these actionable pillars are where we discover and the direct connection of our products and services and processes to sustainability. Nourish, protect, and innovate. Kemin has 6 core technologies which are used to create Kemin products. It's through these technologies that are scientific innovations that we discover and deliver on our commitment to our customers and our mission. Kemin business units deliver innovative products and solutions for the human and animal feed and food industries around the world. Kemin also works in the nutrition, pet food, textiles, and commercial horticulture industries. And you will note the animal nutrition, aquaculture, pet food, human nutrition, food technologies, crop technologies, and textile auxiliaries share a lot of the molecular advancements to be able to serve their individual industry needs. Our animal, human nutrition and health serves the industries around the world. Kemin is strengthening the safety through the food chain, improving animal nutrition to help producers raise healthy livestock, poultry, and equine. We believe animal nutrition and health is a crucial factor in meeting the needs of the animals and a growing population. The Kemin Human Nutrition and Health business unit is committed to providing customers and consumers with new innovative solutions and ingredients. All ingredients are plant-based, clinically studied, and based on consumer health needs. Kemin Human Nutrition and Health is accredited with successfully commercializing lutein and significantly developing the eye supplement category. Knowing the level of lutein and zeaxanthin in your eyes can help take safe and easy measures to help protect and keep them healthy. We have solutions in eye health, brain health, immune health, which Chris Sadawasser will talk a little bit more about after my talk, weight management and sleep. At Kemin, we invest in safety and quality standards that are far above the industry average. This is part of our promise to customers. We act in partnership with our customers to fulfill their needs and expectations while achieving mutually profitable results. We have extensive safety programs, quality, as well as our customer laboratory services. Kemin is an active partner in every community in which we work. We strive to promote the economic and social well-being of the communities and where we are manufacturing as well as living. Kemin believes in thinking globally and acting locally. Our employees support many organizations individually, while Kemin as a company focuses on science, general education, affordable housing, disaster relief, and the creation of vibrant communities. Servant leadership is at the forefront of our values. Every year, employees receive a volunteer day to donate their time to the organization of their choice. Thank you for your time, and I hope you gained a little insight into Kemin.
I'm Chris Sadawasser, a Technical Service Manager for Kemin Human Nutrition and Health. And today I just want to take a few minutes to share some information about beta-glucans. Immune health is a pretty big topic recently. So naturally, there are a lot of questions about immune health ingredients. I want to take just some time to discuss one category of the ingredients called beta-glucans. We will look at what they are, how they work in our immune system, and how we produce beta-glucans at Kemin. So let's jump in. What is a beta-glucan? Beta-glucans are just long chains of glucose molecules that form a non-digestible fiber. Beta-glucan is found in a variety of different organisms, including cereal grains like oat and barley, fungi, bacteria, and algae. The molecule shown is an example of a beta-1,3 glucan. And you can see the two glucose molecules bound together. The carbon labeled 1 on the left glucose is connected to the third carbon on the right glucose. That linkage is what the 1,3 name refers to. Beta-glucans exist in a variety of structures depending on the source. Those different structures impact how the beta-glucans interact with our bodies and when we consume them, and what health benefits are associated with them. But not all beta-glucans are the same. These different sources of beta-glucans can have different health benefits based on their function, structure, size, and solubility. We see that cellulose found in plants is a 1,4 structure and is associated with gut motility. Oat and barley has a beta-glucan that has mostly 1,4 linkages, but also has some alternating 1,3 linkages as well. This structure is associated with cardiovascular health benefits. Those benefits are often advertised on the boxes of whole grain cereals, the Big Heart. The beta-glucans we're primarily focused on today is beta-1,3 glucan, which leads to immune health benefits. These 1,3 linkages are recognized by and activate key immune cells in our bodies. A great source of these beta-1,3 glucans is from an algae called Euglena gracilis. We see this 1,3 pattern in yeast beta-glucans as well, but it has 1,6 branches, and the beta-glucans are also found in the yeast cell wall. That makes it difficult to separate the beta-glucan from a cell wall without additional processing to access that beta-glucan to help achieve a higher purity. Euglena's beta-glucan is not bound up in a cell wall, which makes it a perfect natural factory of beta-1,3 glucan and other nutrients such as protein, essential fatty acids, vitamins, minerals, and some carotenoids. Here's a larger view of a Euglena gracilis. It's a freshwater microalgae that creates and stores beta-glucan as an energy source called paramylon. Paramylon are those little white granules in the Euglena. Kemin grows a proprietary strain of this freshwater microalgae, and it's a strain of Euglena gracilis. It can be grown both heterotrophically or autotrophically. Autotrophic growth is when algae uses the sun to create energy through photosynthesis, a word many of us haven't really thought about since high school biology class. But photosynthesis causes the algae to produce more chloroplasts and chlorophyll, the stuff that makes plants and other species of algae green. This is how plants generate the energy to survive. But Euglena can also be fed heterotrophically, which means it's when we can feed nutrients and minerals without the need for light. When Euglena is fed heterotrophically, it stores energy in the form of paramylon, which is made up of linear beta-1,3 glucan in a tightly wound helical formation. Unlike yeast, the beta-glucan the outer protein membrane is easily broken, and we can release those free-floating beta-glucans, so it's easier to interact with our immune cells. An added benefit of the protein-rich membrane is it contains amino acids that help contribute to the protein content of beta-via complete. So let's take a closer look at the paramylon. As I mentioned earlier, Euglena accumulates these granules, and these granules are made entirely of long chains of linear beta-1,3 glucan. The beta-glucan chains are wrapped really tightly into a small crystalline granule. We can think of it like thread being wound into yarn in a helical structure, and then that yarn is wrapped into a really tight ball of yarn. This tight and organized structure leads to a very stable granule, just entirely beta-glucan. As we discussed before, the beta-1,3 glucan is recognized by key immune cells as a pathogen-associated molecular pattern. That special pattern helps to prime the immune system to help us become better prepared to protect our bodies and to maintain health. Kemin grows our strain of Euglena in a patented fermentation process, which promotes the Euglena to accumulate beta-glucan to greater than 50% of its dry weight. We call this product Beta-Via Complete. Beta-Via Complete is a proprietary, nutrient-dense strain of Euglena gracilis for immune system support, containing greater than 50% beta-glucan, also protein, essential fatty acids, vitamins, minerals, and carotenoids. It also has been shown in a human clinical trial to support immune health in an active, healthy population. At Kemin, we really specialize in natural branded ingredients that are purposely cultivated and extensively researched to promote health and well-being. Throughout all of our business units, Kemin has been committed to promoting immune health, and it is clearly an important topic. Beta-Via Complete is our first immune health ingredient in the human nutrition market. The immune health market is experiencing large growth as consumers continually continually looking for ways to stay healthy. Beta-Via Complete provides a new, natural, GMO-free source of beta-glucan that can be easily incorporated into immune health formulations and functional food. The Euglena for Beta-Via Complete is grown in enclosed tanks using a patented fermentation process. The fermentation process takes place in enclosed tanks to minimize the exposure to weather and other contamination that might occur when algae is grown in like an open pond or a raceway pond. It also leads to a consistent, high-quality product. After fermentation, Beta-Via Complete is then dried and milled to create that whole cell algae powder. This contains all the cellular components of Euglena gracilis, including micronutrients and protein, in addition to that greater than 50% beta-glucan. You'll notice this algae is not green. That's because we grow the algae in the absence of light. We promote the growth of that paramylon while reducing how much chlorophyll accumulates, which would lead to that green color. The absence of that chlorophyll, along with those added nutrients and protein in the algae, is what gives our product that distinct golden color. At 50% beta-glucan, Beta-Via Complete is like the highest beta-glucan content of a whole cell immune health ingredient with a dose of 375 milligrams a day. We currently have a shelf life of 3 years on the product. The beta-glucan is a very stable granule and very resistant to breaking down. This really helps when it comes to formulation. You can typically use lower overages because the ingredient is so stable. And this stability holds up really well even when pH and temperature changes happen, which are common when we're processing into finished products. So with 50% of the product being beta-glucan, what makes up the other half of Beta-Via Complete? Beta-Via is not just those beta-1,3 glucans, but it is also a nutrient-rich fermentate. And we can see a breakdown of a few of the key nutrients which make up that composition and also contribute to the immunonutrition of the product. First, we have the beta-glucan, which we have discussed in depth and is tied to that immune health component. Next, we have protein, which is made up of amino acids. Also contains fats such as omega-3s and omega-6s. That contributes to the unsaturated fat content found in the product. There's also vitamins and minerals present in the material. And last but not least, Beta-Via contains some carotenoids, such as lutein, which Kemin knows a lot about. With all these nutrients, Beta-Via Complete is an all-natural superfood that can help support a healthy immune system. Also, its light golden color and low dose makes it easy to incorporate into a finished product. Beta-Via works well in capsules and tablets and a variety of functional food and beverage applications. With that, I want to thank you so much for your time and attention, and thank you for joining us.
Beta-glucans are a well-researched class of molecules found naturally in pathogenic fungi and bacteria. They play a key role in immune function by alerting the host animal of pathogens, triggering the immune response needed to overcome a challenge. Today, Beta-glucans are used by animal production systems to prime or enhance the protective immune response in animals. The function of beta-glucans and bioavailability differ by the size and type of linkage in the polymer chain. For example, 1,3-beta-glucans are a small, linear class of beta-glucans that can provide a consistent immune response, whereas branched 1,6-beta-glucans are bulkier and can elicit a more variable response. So, how do beta-glucans work? When fed, 1,3-beta-glucans enter the small intestine and are absorbed through Peyer's patches, which are a part of the gut-associated lymphoid tissue. Immune cells, such as macrophages, recognize 1,3-beta-glucan chains through cell receptors located on the cell's surface. Macrophages then engulf 1,3-beta-glucan chains and transport them throughout the body, releasing smaller fragments of the beta-glucan chains along the way. The immune cell's recognition and ingestion of beta-glucans trigger the release of signaling molecules. These molecules activate other immune cells, like natural killer cells and neutrophils, T cells or B cells. The pre-activation of these cells allows them to be primed and ready to elicit a response to disease challenges. The activated or primed immune cells target and destroy foreign cells and disease-causing organisms, minimizing pathogen load. The overall effect enhances the immune system, enabling the animal to overcome disease challenges, reducing morbidity and mortality. Alida from Kemin is a highly bioavailable, concentrated source of 1,3-beta-glucans derived from algae. Alida is designed to prime rather than activate the innate and adaptive immune response, which is particularly beneficial to animals under disease pressure. A quiet immune system may not be prepared for a challenge, whereas a needlessly activated immune system may cause a loss of valuable nutrients that could be used for key functions like growth or maintenance. Alida is optimally used during times of stress or disease like respiratory challenges, in young animals that are developing their immune systems, and in all ages of animals to enhance overall immune efficiency. For more information about Alida, visit kemin.com/Alida-US.
Okay, so that's our first part of our exhibition. So I'll welcome back Penny, Brenda, and Chris. Any other comments from us before we take any questions?
Yeah, no, thank you. And we appreciate the opportunity to be here today and to share a little bit about Kemin and looking at how our ingredients are used in different applications and different industries, which I think is a fundamental benefit of the topic that we have here today and the purpose of this Congress. So we welcome any questions that you might have about what you've seen.
Yeah, if there are any questions, you can unmute yourself or you can type it in the chat and I will read it.
Yeah, I think one question I have is, which I think might be relevant to maybe early career scientists, PhD students, and other early career scientists, is in terms of trying to take products to market, from the laboratory into the commercial space. I mean, is there a chance you could just please provide some highlight on that, on how, drawing experience from the Kemin side of things, and what sort of opportunities are there for scientists here that are working on interesting molecules in their research, particularly. Maybe you can just use a case study from Kemin. I think that would be nice.
Yeah, for sure. And actually, Geoff, if you're open to it, you might be the best to speak to this, since that's exactly what you've done along the whole path here, from having your own independent business to growing and expanding. So, yeah.
Sure. Yeah, I'm happy to take that one. So I think, one of the biggest challenges for anybody that's contemplating bringing a new molecule to the market, and Euglena makes all sorts of very interesting molecules, and I think there's a lot of opportunity to still utilize it as a as an organism that's flexible enough to make some of these molecules. But in order to bring those to the market, it is quite challenging, and there's some important considerations to think about very early on. One, which was kind of touched on earlier, was the GMO nature of it. So yes, Euglena might be manipulated with GMO tactics, and that could limit its ability to be marketed in certain markets. So that's one important consideration. So for our products, we're using our own proprietary strain, but it is not a GMO strain. And so we're just taking advantage of Euglena's natural tendencies to make beta-glucan naturally. So there's the GMO consideration. The second is really more the business side of it. So if you're going into animal health, you'll notice that there's not a lot of snake oil that's being sold in the animal health space. So it's a very, I would call it sophisticated industry, where if you're making claims about any sort of benefit that comes from a molecule, you need to back that up with lots and lots of studies. And so it's a pretty high bar to meet. And so when you go to these industry conferences, you need to be backing up any sort of claims. And so we spent lots and lots of research money just trying to demonstrate the mode of action, which we shared a little bit earlier, but then also just doing actual trials to say, okay, if we think that we're going to help the immune system, show us how that's going to work in a chicken, pig, shrimp, fish, etc. And so it really needs to be very well studied and documented to get into the animal health space. Certainly on the human health side, there's a lot of products that are peddled out there that claim to have immune benefits or whatever the benefit would be. But the ones that have real staying power are the ones that are also very well studied. And so that's what really Kemin is very good at, is recognizing some of those molecules from a very early stage. And so maybe Kemin recognizes a new molecule that's coming up through the research world. They say, okay, how can we maybe make that molecule with a plant that we have experience with? Or how do we actually mass produce this new crop in a sustainable way, but actually bring it to market with the science backing and also navigate all the regulatory pathways. So I'd say that if you're a researcher that's identifying some new molecules, the key thing is you're probably going to need some bigger partners to help do that. And so a company like Kemin, 3,000 people, we've done this before. There's 500 other molecules in the portfolio. And so, I think that the best advice for me would be try to partner up with somebody that can navigate some of those things, and you can focus on what you're good at, which is identifying a new molecule. And then other companies are good at navigating the next steps, which includes regulatory. And at the end of the day, you also have to sell it. And so having a sales team that is already in the marketplace for whatever target for your molecule will be, that's an important consideration to have a successful launch.
Yes, so we have a couple of questions from the chat. One is from Ellis O'Neill, and the question is whether the proprietary strain that you have developed, has that undergone much breeding, and what technology do you use for that breeding, and whether it's all aimed at total biomass or a change of distribution of the nutrients. So whoever wants to.
Once again, Geoff, maybe you'd be the best here to answer this one.
Yep. So very simply, we haven't used GMO technologies to do the breeding for this Euglena gracillus strain. It's convenient in this regard that if you grow Euglena at maximum speed in a fermenter, it also generates a lot of beta-glucan. So we got lucky, maybe not lucky. Yeah, maybe it's lucky. So the same growth strategies that will maximize growth also maximize beta-glucan content for us. And so I would say that our challenge was a lot easier than if you're trying to target a molecule that's only produced under stressful conditions or under certain triggers. And so, so in this regard, yeah, we didn't do anything overly exciting on the breeding side of it. So it's, not to say that we didn't put a lot of effort into it. Clearly, a fermentation tank is a whole lot different than the natural ponds that these Euglena are naturally adapted to. And so a lot of it was adaptation to our growth conditions as opposed to selection for for beta-glucan.
Thank you. And then there's another question from John Knight. Does Kemin use Euglena gracilis for other products besides beta-glucan?
And I can take this one, Geoff, if that's okay. And at this point, we use our Euglena gracilis for just for beta glucans. So we use it. We have two different varieties. One that is the full algae fermentate, and one that is more of just the purified beta glucans. But we are very happy with working with the algae. We feel like that's a mechanism for producing other molecules in the future. So that's something that we're quite interested in. We feel it's a sustainable source for producing ingredients. We definitely like to produce ingredients using natural sources. And so that is something that we look into for future. But at this point, we just have the beta-glucan products from our Euglena gracilis.
Yeah, maybe the last question. What is the oil content in your biomass, and have you tried to determine the type of lipid, TAG versus wax ester?
Geoff, do you have that? Or Chris?
I can speak to that a little. Geoff can probably correct me, but so we really maximize the beta glucan content. So the oil content is pretty low, less than 10%. So we really haven't focused a lot on doing that. I think we have looked at the breakdown. I don't know it right offhand, but definitely our fermentation is fine-tuned to kind of maximize that beta glucan content. Really, in order to do that, we minimize a lot of the other components that are being produced, like the oils.
Okay, so thank you very, very much. And I would suggest that we move on to the next presentation by Noblegen. And maybe I can call Scott to share the video.
I'm back. Thanks. That was a really great presentation. Okay, so I'm going to share our showcase, and it's about 33 minutes long, and then there'll be time for a Q&A at the end as well. Okay, so I'm just going to share my screen. We'll get started here. And again, we're going to share the video on YouTube afterwards, if you'd like to watch it on your own time.
Thank you for having us at the first annual International Congress on Euglenoids. We hope all of you have enjoyed the conference so far. We don't have a lot of time, and we have a ton of material to share today. So without further ado, here's Noblegen's Industry Showcase. 2020 to 2021 has been a year of substantial behind-the-scenes growth for Noblegen. Navigating our way through the COVID-19 pandemic hasn't been easy, but it did give us the opportunity to take a step back and look at the bigger picture of the company. As a result, we've laid the groundwork for what Noblegen is now and what Noblegen will be in the future. Noblegen is more than food and ingredients. Noblegen is science and innovation. Here's a quick snippet of what we've been up to.
We are scientists. We are innovators. We are Noblegen. At Noblegen, we work symbiotically with Euglena to solve some of the food and beverage industry's biggest challenges. We grow and cultivate Euglena food ingredients and meat analogs at our world-class fermentation facility in Peterborough, Ontario, Canada. An ancient process, Euglena evolved before the distinction between plants and animals, able to switch between photosynthesis and organic carbon ingestion. Noblegen has unlocked a natural way for Euglena to convert organic carbon into solid tissue using a 20,000-liter bioreactor with a rich, smoky flavor, just like animal muscle tissue. No additives or extra processing required. Nutrient-rich, sustainable, a complete game changer. Our first meat analog is our texturized Euglena protein. It mimics the look, texture, and taste of chicken at a fraction of the cost and resources. Real vegan meat is coming, and Noblegen is leading the way.
Noblegen is located in Peterborough, Ontario, Canada. We have a few different locations across Peterborough. Our headquarters is located downtown. We have a lab and office space at one of Canada's best universities, Trent University, and we have our Gen 1 fermentation facility in the industrial area of Peterborough. Our facility, pictured here, has a fermentation capacity of 160,000 liters. It's one of the largest facilities of its kind in North America. It has an in-house R&D workspace, as well as a fully equipped in-house lab. It also has in-house downstream capabilities. Here's a video tour of our Gen 1 facility to give you a glimpse into what our current facility is capable of.
Welcome to Noblegen, where we create nutrient-dense products out of naturally occurring Euglena grassless with minimal processing. My name is Callie Mitchell, and I'm the operations manager here at our Peterborough-based SQF certified R&D and manufacturing facility, where we have a 160-liter fermentation capacity. That's 8 of these massive 20,000-liter fermenters. But we don't start there. We start here by inoculating these flasks with Euglena grassless cells and our proprietary feed recipe. They grow and grow until they can be moved into our larger production fermenters. Then we use our pilot-sized fermenters to continue feeding and growing our culture until it's at a volume large enough to inoculate a 20,000-liter fermenter. Throughout the life cycle of a fermenter, we monitor and analyze different features and factors like pH, temperature, cell density, the composition of the cells, and more. For example, every 24 hours, the Euglena will double in size. These pieces of data allow us to make informed decisions about our daily activities and how to maintain the healthiest homes for our cells. Our facility is equipped with high steam capabilities, which allows us to heat and sterilize our piping and equipment. From the DCS behind me here, we can send feed, water, and steam where we need it to go, control temperatures, monitor various process parameters, harvest, and control equipment. Once our cells have grown to optimal volume, density, and profile, we attemper the cells to halt growth, separate the water and media from the cells, and we're left with our biomass. Our spray dryer allows us to create and package in-house final samples of our current products, as well as products under development. Bulk volumes of biomass are dried at a third-party facility equipped for large-scale drying and packaging. Biomass can become any number of final products. We can separate the protein and beta-glucan and dry them into flours, or we can make a liquid vegan egg, whiteners, and creamers, or even extrude it into a meat analog.
Our Gen 1 facility is only the beginning for us. Noblegen's next phase of growth will include building an all-encompassing facility in Peterborough that brings all of our departments together under one roof. Our Gen 2 facility will be built in Canada's premier clean tech destination, the Clean Tech Commons at Trent University. The Clean Tech Commons is a Canadian hub for collaborative clean, green, low, zero carbon, and sustainable technology research, innovation, commercialization, and entrepreneurship. Construction on our Gen 2 facility is likely a couple of years out. In the meantime, we have been in the process of securing fermentation tolling assets all over the world to strategically produce our ingredients closer to the markets we're planning on entering. The versatility of Euglena's feedstock frees us up to bring our technology anywhere in the world. Not only does this allow us to bring our food, ingredients, and tech to the people that need it most, but this approach will allow us to keep our carbon footprint as minimal as possible. Speaking of the people that need our technology the most, Noblegen, in collaboration with Safe Haven Solutions and Producers Market, will be a part of the UN-backed Grand Bahama Agri-Park that is being built with aims to reduce import dependency and to provide food security for the Bahamas. The Agri-Park will be sustainable and resilient using the Sustainable Development Goals established by the United Nations and the 25% by 2025 plan by CARICOM as its focus. It will also be the first of a series of agri-parks that are being planned in strategic locations around the world to provide food security to nations in need all over the planet. The agri-park will provide locally grown organic produce for the Bahamas and CARICOM and will be powered by renewable energy. The project promotes sustainable living and will have a positive social, environmental, and economic impact on Grand Bahama, the Commonwealth of the Bahamas, and CARICOM. We're excited and honored for Noblegen and Unite Foods to be a part of such an amazing project. Unite Foods is our food and ingredient brand. All of our food and ingredients will fall under the Unite umbrella going forward. That frees Noblegen up to be the science and research foundation of the company. As you can see here, we're calling it corporate mitosis. Unite is a modern agricultural food brand that is reimagining how the world eats and working to re-evolve the food system as we know it today. Using the power of Noblegen's microbial platform, Unite efficiently produces patented protein, carbohydrate, and lipid ingredients, as well as a full complement of meat, dairy, and egg analogs that cover all aspects of nutrition and functionality, while providing an unparalleled ecological advantage in comparison to other ingredients and animal analogs available today. With Unite, our goal is to build a food and ingredient brand that is recognized as having nutritious and delicious food and ingredients that are synonymous with sustainability, food security, and climate action. We're pitching ourselves as the Intel inside of food. If our ingredients are in a major CPG's product, then it means they're willing to provide their consumers with sustainable food options that actually help the environment. Our goal is to make the Unite Foods brand mean something. And to do that, we need to be open and transparent. We need to educate consumers on what Euglena is and what its benefits are. And we need to start a community of early adopters that are willing to work with us to improve our ingredients and food products throughout the process. The wheels are in motion to start all of that as soon as possible. As for our food and ingredients, as you can see here, we have a lot going on. Our base, or V1 ingredients, are our dry ingredients: Euglena protein-rich flour, Euglena protein concentrate, Euglena beta-glucan isolate, and eventually Euglena lipids. Our V2 ingredients are our functional ingredients: texturized Euglena protein, or TEP for short, Euglena protein crisps, functional beta-glucan isolate, and eventually solid Euglena fats. And our V3 ingredients are our Euglena food systems: egg, meat, and dairy analogs. We're confident that we have one of the most robust and complete product pipelines in the industry. There are a ton of different routes we can potentially go in, whether it's B2B, D2C, or white label licensing. Anything and everything is currently on the table. Here are a few images of some of the food demos our product development team has put together to date. Not pictured here are tons of other interesting finished foods made with Euglena. TEP fish sticks and animal-free chicken noodle soup, animal-free beef burgers and crumble, dairy-free yogurt, creamer, and mac and cheese are all demonstration products our team is currently working on. While we're on the subject, here's a food demonstration video we put together with our innovation chef, Loren Cameron. Next year, we're hoping we'll be able to do a food demo for all of you in person so that you'll actually be able to taste food made from Euglena. Until then, this is the best we can do. Enjoy. Hi, everyone. I'm Brian Reed, the head of communications and media at Noblegen. Today, we're going to head on into our innovation kitchen with our innovation chef, Loren Cameron, and she's going to walk us through a whole bunch of food demonstrations. So without further ado, let's head on in. Welcome to Noblegen and Unite's Innovation Kitchen. Today, I'm with our Red Seal certified innovation chef, Loren Cameron. Loren's going to show us how she prepares our texturized Euglena protein in different food demonstration dishes from start to finish. Thanks for having us here today, Loren.
Yeah, thanks for stopping by.
Awesome. It's really great to be here in person and to do this kind of thing again. So before we get into the food demonstrations, we're going to get into a little bit of an explainer on how we make the texturized Euglena protein. So at Noblegen, we have our own plant where we create or cultivate, I guess, and grow Euglena in fermentation tanks. That is eventually cultivated and brought out in a liquid biomass form. We add a little bit of pea protein to it, and then we send it off to our extruders for them to do their thing and extrude the product. So to start, we're going to go with our strips. And as you'll see, they're very meat-like. Like it almost looks like these could be cuts of meat, cuts of cooked meat. So again, we're gonna show you right now how great this stuff is at tearing and what it kind of looks like as you're shredding it and how it mimics animal-like muscle fibers, basically. So if we have a peek here, you'll see how it tears apart there. It's got the fibers that kind of keep it connected, and yeah. So it's pretty cool. And Loren's going to prepare a dish for us today. And how are we going to go about that?
Yeah, so as Brian kind of just explained, it tears really nicely. And so when I first started working with this, the first thing that came to mind was pulled pork. So the first dish we're going to show is a pulled pork taco. All right, so before we get started on the pork, I'm just going to put together a quick fresh salsa. Thing about this is it takes on the flavor very easily of what you're cooking it with. So it's a very kind of neutral type natural flavor that it has. So I love working with it because it just picks up the flavors so easily. So all I'm going to do is I add a little bit of oil to this pan. I'm going to add some of our pulled pork and let it heat up a little bit. So that's also one of the reasons we can use it in so many different applications. We can use it for pork, we can use it for chicken, we can use it for beef, just because it takes on the flavors that you're cooking with it. And here at Noblegen, we really love fermenting things and kind of playing with different foods. So a little while ago, I actually fermented some ketchup for the staff here, and I still had some in my fridge, so I actually just whipped up a quick barbecue sauce to go with it. So it's a nice kind of smoky, sweet with a little bit of spice sauce. It's gonna go perfect for our tacos and really complement the freshness of the salsa that we're gonna put on top. And give it a little bit more of that meat texture. And all we want to do is kind of heat it up to that 165. We're not really looking to kind of cook it all the way through. We just always want to meet that mark for anything that we're reheating. All right, that should do it. So we're ready to plate here. Like I said, I'm kind of pretty simple. So we're just going to throw a little bit of the avocado vegan mayo straight onto our corn tortillas. Add some of that pulled pork that we just cooked up. It's nice and saucy.
Looks really pulled porky.
Mm-hmm. There you go, some fresh salsa. Now, are you a cilantro person or not a cilantro person?
I am not a cilantro.
No cilantro.
But
Right.
you're Red Seal certified, so I'll trust whatever you want to do.
All right, well, we'll throw a little bit of cilantro on yours, and then I quite like cilantro, so we'll put a bit more on mine. Nice little garnish, a pop of some green there. And then, yeah, I think we are good to go, guys.
All right, shall we eat? Let's give this a shot.
My mouth's been watering, so here we go.
Look very pulled porky. I'm very excited. It smells delicious. It's good.
Mm-hmm.
I do like the sauce, the fermented ketchup sauce.
Mm-hmm.
It's really nice.
Got like a nice little bit of heat.
Look at it though.
Mm-hmm.
That looks just like pulled pork. Tastes like it. So the next dish we're going to move on to show you guys the versatility of the TEP. So next we're going to do a chicken dish. So with the chicken dishes and some of the other dishes, we generally use these chunks of the TEP. So it's dehydrated. You can see it kind of looks like a pork rind. So all we really have to do is add a little bit of liquid to it. It brings it back to life, and it tastes like chicken and looks like chicken. So I'm very excited for Loren to show us the next step. What kind of dish are we gonna do with the chicken?
So we're gonna keep it pretty simple, and we're gonna do a chicken stir fry. So just some fresh veg in there, a little bit of stir fry sauce, and put it on top of some rice, and let the TEP do its thing. To reheat the TEP here. So like Brian was saying earlier, it's really simple to rehydrate. All you have to do is add some liquid to it. The nice thing about it is once it's done taking the moisture it needs, it doesn't take any more. So you can simply kind of get it to the moisture you want and then drain out any extra. So it also really takes on the flavor of whatever you're adding to it. So I have some actual like vegan chicken broth here that we're gonna pour in. That should be good enough. And give it a little stir. And then we can move on to start to cook our vegetables. And by the time we are done that, this will be good to go. Usually it takes about 5 minutes or so for it to fully rehydrate. But again, because it doesn't take extra moisture, once it's done, it doesn't need to be drained immediately. So it can sit for 10-15 minutes while you're doing something else. And then you can get back to it and drain it. All right, there we go. ever drain TEP here, and I'm just gonna add it straight into the pan, let it heat up for a little bit, and then add the sauce in.
All right, so the Euglena texturized protein chicken stir fry is done, and Loren's just gonna plate it for us. It looks great. Can't wait to eat it.
Yeah, so we're just gonna throw it on top of some jasmine rice here. Get some of those fresh veggies and chicken going on. There you go. Finish it with some sliced green onions. And there you go.
All right, let's give it a taste. Looks good. Can't wait. Try to get a big old chunk of it. The TEP. Forks are hard.
All right, first impressions. What do you think?
Surprise, right? Again, it looks just like chicken. I mean, look at this. Looks just like the chicken, tastes like it, similar texture, everything. It just feels bang on. I can't believe how close it is. I just want to keep eating it. All right, so we've had a pulled pork dish, we've had a chicken dish. So we're gonna move on. We're gonna show you guys a little bit of what we can do for a tuna dish. So you've already seen the strips that we've used and the chunk that we used for chicken. Well, here's the flaky texturized Euglena protein that we use for tuna. So basically all we do, again, is add moisture to this, and it'll take on the flavor of whatever moisture it is, and then we'll put it in a dish, and then we'll give it a taste.
All right, so as Brian mentioned earlier, I'm working with our flaky TEP. So this is quite similar to our more chunky chicken style TEP. It rehydrates really simply just with a little bit of water. For this one, actually, I did something a bit different. I rehydrated it earlier with a little bit of liquid smoke and a seaweed reduction to give it a bit of a smoky tuna flavor. I actually then re-dehydrated it, and so now all we're gonna do is add a little bit of water and some other seasoning in it. So again, it kind of just takes what it needs, and this one rehydrates a lot faster, so you can kind of just give it a stir and wait until it gets that consistency you want. You can already see it starting to come together and really getting that tuna look. Now, since I'm adding a little bit of an avocado vegan mayo, I'm not going to let it rehydrate completely with just the water. Go in and add some of this in now. All right, so that's starting to shape up to kind of the consistency that we're looking for. For that, I'm going to add some green onions. And that's quite literally our tuna salad. All right, once again, guys, we're keeping it pretty simple. We're just doing a classic tuna salad wrap. Put some lettuce down there. Some of our smoked tuna mixture. And then just wrapping it up.
Smells like a smoky tuna. Looks like a smoky tuna. See that? That nice little texture, little tuna melt like grandma used to make. Love it. I was gonna throw that away, but. Tastes like a flaky tuna. I mean, just the same consistency. I like the little flaky chunks. It's nice, the smoky flavor you did.
The texture is spot on with this one.
Mm-hmm. Yeah, it's not, sometimes it can be a bit chewy, but with the flakes, it just kind of dissolves in your mouth. It's really nice. Cool. Well, thanks, appreciate it.
No worries.
Well, Loren, I think we've taken up enough of your time today. I really appreciate you making the time and making all this great food for us.
It's always great having you guys here in the kitchen and getting to show you what's happening at the Noblegen kitchen here.
Awesome. I think you nailed everything. I mean, the chicken tasted like chicken, the pork tasted like pork, the tuna tasted like tuna. Everything was really great. And I want to give you and the product development team out of Minneapolis a lot of credit for how far we've come with all of our products. We've come all the way from Marini tasting powders to finished food products. And I really honestly can't wait to see what you and the rest of the team can come up with next. But until then, thanks for watching. And we'll catch you next time.
Bye.
It's too bad we weren't able to share all of that in person. It truly is amazing how far our product development team has come in a very short amount of time. So that's what Unite Foods is all about. Let's get into a little more detail about the direction that Noblegen is going moving forward. Noblegen's strong scientific foundation led us to the food and beverage industry, just as it will lead us to other industries. We've always explored microorganisms and learned from them to try to imagine tools or products that positively impact the world because we believe that microorganisms can provide unique solutions to real-world problems. We've been able to develop an entire food system based on Euglena. We're now looking at how Euglena interacts with other microorganisms and working to discover its true potential across industry. Outside of food and agriculture, health, water, biomaterials, energy, mining, and space are all industries that we're confident can utilize our Euglena platform. In fact, we were just named finalists for the Deep Space Food Challenge. Still the food and agricultural industry, but also space. We're pretty excited about it. Before we show you a quick teaser video we submitted for the challenge, let's get into a little more detail on what the Deep Space Food Challenge is. As you can see here, the Deep Space Food Challenge is a joint effort between NASA in the United States and the CSA in Canada. The challenge is to create novel food production technologies or systems that require minimal inputs and maximize safe, nutritious, and palatable food outputs for long duration space missions that also have the potential to benefit people on Earth. The drive to develop space technology for low mass, low energy, and low waste systems is directly applicable to the goal of harnessing space to solve everyday challenges for Canadians, in particular for Canada's northern and remote communities. The same can be said for remote regions with limited resources all over the world, something that Noblegen is truly passionate about. We think Euglena is the perfect food source for this challenge. Unfortunately, we can't show you the entire video that we submitted for the challenge, but we can show you the introduction as a teaser.
Biological life support systems offer a unique solution to sustaining astronauts in space and are seen as an integral part of deep space travel. These systems leverage the biology of robust organisms tuned for efficiency and survival. Our plan is to use safe microbes to upcycle human waste into nutrient-dense feedstock for our platform organism, Euglena gracilis. Euglena is a robust, single-celled eukaryote with a dynamic metabolism and amazing nutrient profile that we're already using on Earth in food applications. Our Euglena biomass is GRAS safe for consumption, and by engineering it for taste, texture, and smell, we will provide a unique and healthy food solution for deep space travel.
We hope you enjoyed that quick snippet, and we look forward to sharing more in the future. That's all we have for our update today. We hope you enjoyed learning a little bit more about Noblegen and everything that we have going on. 2022 is shaping up to be a year of massive growth for Noblegen. As we work to bring in our Series C funding, we'll be looking to grow and expand our team. If you're interested in any job openings or Noblegen updates, be sure to follow us on our social channels and keep an eye on our careers page. We'll be posting any and all company updates there. If you want to collaborate with us on any specific projects we're already working on, or if you have a project in mind that we could potentially help with, we're always game to hear some ideas. Feel free to email us a pitch at info@noblegen.com, and we'll see where that takes us. Thanks again for taking the time to learn about Noblegen, and thank you to the EIN for organizing and hosting this event. We can't wait to see what kind of collaborative efforts come out of this network going forward. Thank you.
Okay.
So, ready for questions? So, I mean, it's getting dinner time in Central Europe. So it was really, seeing all those things was both, I mean, I hope we are going to be able to taste all of that soon. So, are there any questions from people who wanted to ask live? Then I can try to extract some from the chat, but...
Yeah, I think most people are asking via the chat box, so maybe it's something.
Yeah, so there is a question about the paramylon beta-glucan content and whether this can be eaten long-term.
Yeah, so the paramylon is, as we know, the granule that's comprised of majority of beta-glucan. So far, we see no, and as Kemin echoed, you can eat it, a lot of it, without any, you have the immune boosting effects. So I don't see a limitation to using it in products. I don't see a limitation to eating it.
Yeah, I think there was a follow-up of the question saying, if beta-glucan induces immune response, is it healthy to eat it in large amount for extended time? That was maybe a clarification of the question.
I think there's clinical studies going on to figure out how much you can eat over a long period of time, but so far we haven't seen any limitations to it. But Kemin might have some comments on that.
Just real briefly, we have done long-term human consumption studies, but only at the dose levels that are at the optimum levels for the immune response. So we've given it to animals for a very long time, and for humans for 3-4 months at a time. So yes, you can do it for long periods of time, but at very high dose levels, that's not something that we've done for extended periods of time yet.
Yeah, I don't see other questions in the chat, but I have a question myself, maybe to Scott. So we have seen already a little peek in the future with this space food challenge, but would it be possible to have a more extended peek on, where do you expect to see big progress or big growth for Noblegen in the next, say, 4 or 5 years?
Absolutely. Food was our major initiative to start, but Noblegen was actually founded on wastewater treatment technology and how to remediate kind of soils and wetlands that have been affected by mining activities. So we see ourselves plugging into that. We have some technology that we've developed for reclamation of those sites. We also see ourselves as a biotechnology company. So we're heading into spaces like the nutraceutical areas. We're heading into areas where we want to see if we can, use Euglena and its remarkable abilities to create new ways to make therapeutics, to make small molecules, etc. So those are just a few of the places that we're going. We're also building beyond Euglena within Noblegen, and we have a vision to use not just Euglena, but other strains and species of Euglenoids and microbes in general to combat a lot of the global problems that we have today. So microbes, as many of us are aware of, they contain, they're each a unique cell factory. They can make a lot of different compounds. They can make They're being leveraged in synthetic biology applications. And Noblegen wants to be a leader in that space as well.
Yes, thank you very much. So I don't know, time is maybe up to wrap up. We can pass you on the leading of the session to you, Scott, and then.
Okay, great. I'll take over. Thanks a lot. So I think the first thing we wanted to do, this is obviously our first go at this international conference. And we wanted to spend a couple minutes just to get feedback from participants to understand, you know, maybe how we could improve things and what you kind of liked and disliked about the online conference. So let's open up the floor to everybody, and just if you have any comments, we'd love to hear them. And I guess maybe the easiest way to do it is just raise your hand in the reaction with the reaction tools, and we can go along and
you can also type as well if you want to type.
You know, I think the conference, the online conference worked fairly well. It takes a little while to adapt to the new platform. Of course, it is different than being in person, so it's hard to recreate kind of that social element. I don't know, I thought spatial was okay, but I also thought, you know, there may be a way to improve the poster sessions, but I don't know, maybe somebody knows of a different platform that isn't prohibitively expensive that we could explore. Any ideas? I think something we can do as well is I can send out a form if people have a little bit of time to digest their thoughts, and we can get some feedback electronically as well.
Yeah, I think that will be better, where we can have like a Google form for people to fill out things, basically.
Absolutely. So I can prepare that and send it out to all the conference participants. If there isn't any kind of feedback on the floor here, we can pivot. And I guess I'll use that time to introduce the plans for next year's conference. So obviously it's hard to predict what's going to happen within the next year, given the state of the pandemic, etc. But we did want to try to have at least a portion of the conference in person. I think, you know, that offers us opportunities to get products out, to sample things, and just create more of a social kind of atmosphere. So the goal is to have the conference in Toronto next year, but as Barry alluded to, we have to reserve accommodation, we have to reserve a venue for that, and that's going to be contingent upon us committing a certain number of people to attending in person. So I'm not going to put the pressure on everybody, but maybe we can just take an initial poll with the audience here to see who would be interested in attending in person. And if there's a palette for that, I'll also send up a follow-up form if you feel like answering that confidentially. But I'll open up the floor to everybody, and maybe we can just get a show of hands or type in the chat who would like to explore a hybrid conference or in-person conference.
Yeah, Ingmar says he purely likes online. So
I saw a thumbs up from Ross and Michael, BJ, Victoria. Yeah, so it seems like some people like the idea of having an in-person conference. That is going to depend on how things improve. I really like the idea of having a hybrid conference where we can have both a virtual and an in-person session. Okay, well, thank you for the feedback. I'm also going to send out a poll so we can get more concrete numbers, and that'll help us in planning for next year. I know I'd love to have host people in Toronto. It's a great city. If you haven't been here before, there's lots to see. It's close to the University of Toronto. It's right on Lake Ontario, so it's got a beautiful view as well. Lots of museums. So we'll see if we can make that happen. If not, we're happy to host online as well. Okay, so look forward to the feedback poll coming in your inbox, as well as the poll for the conference style that you'd prefer. And I'll hand it back over to Thank God.
Okay, thank you, Scott, and for actually sharing this, and to Antonio for sharing this, and to all the presenters, the keynote speakers and the invited speakers, and the oral speaker as well. So we are going to do just one thing that we skipped, which is the poster session, the photo contest. So the idea again is that as part of the conference, we can have very nice poster photo of Euglena, have an award session basically. And again, there are a few things we wanted to do this time, which it now boils down to having dedicated teams, working on this sort of thing, like, oral presenting and so many other things. So I'm sure that Scott and his team would fit into this into the next year's conference, award for oral speakers and all of those, and for poster presenters. So I think I would just like to ask Rosina Sanchez and Takahiro. I think I forwarded the emails. Could you please just let us know who, based on the committee, because just to provide a context to this, we had a call for photo session and asked people to submit their best Euglena photos, either in the lab or in the field. And we had quite a number of submissions. So maybe Rosina will tell us the total number that was submitted, the number that they think is the top 3, and then the number that is the top, the best based on the votes. So there are 3 members of the committee: Macfield, Takahiro, Ishikawa, and Rosina Sanchez. Basically, Japan, the UK, and Argentina, and Mexico. So Rosina, over to you, please. And just to mention that Noblegen is sponsoring, as part of this photo session, an award for the winner. So maybe Scott, you can maybe decide how you want to get in contact with the winner. Over to you, Rosina and Takahiro.
Thank you, thank God. Apologize, the committee was not ready to present the winner, and I had noise issues at my place, but I was telling that we had discussion about the possible winner because there are really good pictures. So let me show you the different photos from participants. First photo is from Jiangxin Huang with photo 1. Again, Jiangxin Huang with photo 2. Again, Jiangxin Huang for photo 3. Jiangxin Huang with photo 4. By Marno Lobster with Euglena sanguinea. Image 1 from Mafusur Shah. Heterotrophic Euglena by Sergio Guerrero. Autotrophic Euglena by Sergio Guerrero again. Euglena gracilis by Michael Hammond. Mix of Euglenoids by Boucina sacris. And that's all. So the 3 finalists were Euglena sanguinea by Marno Lobster, Euglena gracilis by Michael Hammond, and mix of Euglenoids by Boucina sacris. And the winner is a mix of Euglenoids by Boucina sacris. Congratulations, Boucina. And that's all by the Committee of Photo Contests in Euglena International Network. Thank you so much.
Thank you, Rosina, and Takahiro and McField for the assessment. So, Scott, how is it going to happen? I'm not sure how this should work. Maybe in the next year we'll have a better way to do this. So, are you going to get in touch with Bosinia for the award? I'm not sure how it's going to work, but I think we mentioned about an award for them, right? So maybe this is something you can do after the meeting.
Can I say something?
Okay.
Thank you very much to the committee for the award. I really appreciate that, and I'm really glad to get the award. As a taxonomist, I take a lot of photos and videos to document the morphology of studied species. But this is not the only reason. The most important reason is that I just love to take pictures of euglenoids, which in my opinion, and also probably in your opinion, are the most beautiful microorganisms. So I have quite a lot of photos and try to share them with others. That's why you can find them not only in my taxonomic publications, but also in the publications of other authors. I also post them in algae-based, where you can find my photos of species after taxonomic verification, so you can be sure that they show only properly identified taxa. To sum up, if you need photos for your publications of presentations, you just let me know. I will be very happy to share them with you. At the end, once again, thank you very much to the committee. I really appreciate the award. Thank you.
Thank you, Bosinia. Congratulations as well. So I know this is more because this is the first conference, and the number of people organizing this is very lean, and we also through volunteerism. So I hope that from next year things will be more structured, basically as we prepare to formalize in. So Scott, how are we? I'm not sure. Do you want to comment on the award or?
Yeah, I think all these pictures were great. Thanks for submitting them. It's something that will grow as we continue as a community. To actually receive the award, because we can't do it in person, I think the easiest way to do it is the committee members can put me in contact with the winners, and then we can arrange for the award to be sent to them electronically.
Yeah, exactly. So do we need to know what the award is, or is it?
Yeah, the award is money.
Wow.
Yeah, and the award is 500 Canadian dollars. So congratulations.
Thank you very much. Once again.
Okay, without further ado, thanks, Lois, Bosinia, and everyone. I would like to invite Ellis O'Neill to kind of give a closing remark while I support afterwards. So Ellis O'Neill is the vice president of the network, and basically, because again, Ellis, do you want to just chip in something before we wrap up? I know we're keeping people like 15 minutes or so, but we just I hope people can.
Yeah, no, I'd just like to say it's been a fantastic conference. I wasn't involved in organizing it, and I know the committee put a lot of time into it, and I'd like to thank them for that and thank all the speakers. We've had a great few days talking about things that we need for the Euglena research, so more sequencing, more diversity, more studies into it, but also what we've achieved already with identifying the population dynamics of them, where you can find them, how they've evolved, and then today what we can do with them. So huge potential uses for the Euglena. And so thank you to all the speakers, all the chairs that put their time in, the committee that organized all of this, and particularly to thank God, who's been really pioneering this community, the network, and this conference. So if we can put our hands together, thank you to everyone for setting this wonderful week up.
Thank you, Alice, and everyone as well for participating. Thanks to Elam Institute, Nehoa, who has actually been supporting this, and thanks to Rose Law, who is always happy to respond to emails. Thanks to Emily, who basically helped to set up the Zoom platform and ensure that everyone gets in at the right time. Rob Field at the Manchester Institute of Biotechnology for providing to support staff to support all of this, because we basically have our full-time work, but we need people to work with us in terms of volunteering to do this. Then thanks to Noblegen for actually sponsoring the special session, the award session, and ensuring that the Euglena network is also being supported. So just a bit of maybe something we might want to look at for next year, Scott, is that one of the things I've discussed with some members of the community in the past is there are people who have made huge contribution to Euglena over the past 30 years or 40 years. So the idea was to see how we can, each year we have a recognition. I'm sure people who have managed community might know a better way to do this, but that would be a good way to recognize people who have made huge contribution in the science of Euglena. So maybe it's something we need to look into next year. And also for this conference, we try to have, because in the last meeting we have quite a lot of people. Then in this meeting, we try to ensure that people who presented last time, we kind of have new set of people. So as you can see, more than 80% are like new areas. Ingmar wasn't there the last time, and we kind of see the aspect of things, the kind of work he's doing with Euglena, also with many in the area of robotics engineering and so many other things. So I'm hoping that the next conference will be interesting as well. And thanks to everyone for coming. And have a good evening if you are in Central Europe, or morning, afternoon, wherever you are. If there's no other question, I would like to say thank you and see you next year.